Mi sentite? Can you hear me? Can you hear me? Perfect. Unmute yourself, Ale. At least someone on the other side should
Can you hear me?
Yes, but if I mute I think, okay, now it's fine.
Can you hear me?
Yes.
No,
Unmute yourself, Ale. At least someone on the other side should
Can you hear me?
Yes, but if I mute
Pronto, Gioele? Io veramente non sto su YouTube, sto su Zoom. Il fatto che io sento la mia voce tipo molto dopo aver parlato. Voi mi state sentendo ora? Ok, perfetto, ok, per quello. Ora posso parlare, vedi se è sincrono. Sto parlando ora. Ciao.
Aspetta, non sentiamo niente. Ah no, sono io.
Ecco, mi senti orale? Perfetto, allora siamo... Ok, non ci sono stranezze, perfetto, io sto qua. Ottimo. Hi, Martin.
Hello, how's it going?
Nice. Just you are 30 minutes, 40 minutes early.
Yeah, well, it's not that I don't do other things in parallel, so don't worry.
No, it just you might actually listen us while we try to set up things, but that's okay.
Everything going good?
Yeah, things good. What
Great
about you?
stuff with the 3D work of
Thank
the
you so
brain.
much.
I liked it,
Listen,
yeah.
did Adam already join your lab?
Yes.
Cool. Say hi to
I'm
him.
I will. I'm super happy. He's a really good guy.
Yeah. Thorsten says he's amazing.
He is. Yeah. He finally figured out how to do helic LCMS.
Oh. Good. Not an easy thing. And how are things otherwise?
Hmm?
How are things otherwise?
Oh, well, going pretty well. Yeah. I mean,
Are you
busy,
coming
busy,
at some
busy.
point? Are you coming at some point in Lausanne visiting Juliana or?
Yeah, I should. There's also rather regularly with some PhD students there is these committee meetings, but yeah, it's so busy lately that and I was traveling so much that I really did everything online. Shame
Oh well,
on me, but
Yeah, but next time you come, or even if you're
I
not.
will let you know. No
Yeah,
worries.
great.
Yeah. Yeah.
Thank you so much.
Okay, I'll do some other stuff. You call
No,
me
go. Do whatever you want. Just be
when
back
you need me.
be back on time.
Sorry to interrupt. Welcome again. We are going to start the webinar in some time, but we are already in recording and live on Zoom on YouTube.
Yeah. Well,
Just
Okay.
to let you know.
no secrets were shared.
Okay.
Thanks
Now
for
Adam knows what we think about him.
Thanks.
Okay.
Thanks.
See you in a moment.
Bye.
Hello, can you hear me? Audio test.
Yes, Joelle, we can hear you.
Hello? Testing the audio here.
Yeah, this is perfect.
Just to check that you can put speakers on this.
Ale, what is the problem with the slide when I show it?
Let's do another trial, Giovanni, if you can share the screen so that we can try. Okay, you are not in presenter mode.
Pascal,
You should, yeah,
610.
no, still. I think you can just put the
I'm trying
The
to
full
switch
screen
displays,
and
but it doesn't do it.
instead of sharing the presentation, you can go full screen and share the screen directly.
But that's strange. Why doesn't it? Okay. Let me see.
Giovanni, now we see the Illustrator part.
I know.
Okay.
Trying to find way out. No, but it's still Illustrator, so no way. Wait a second. We're gonna get there. Still wrong? Ale, is still wrong?
Okay, so now we see the, no, it was fine.
Was fine?
Try, yeah. But exactly, so like this is okay. We see the whole screen with the background, like with the, properly.
Okay, good.
Yeah.
Yeah, I'll leave it on.
Perfect. Giovanni, still we don't see the full.
We don't see
some of us that is sort of, we show our face to see that there is somebody to talk to. I mean, that's
Hello, everyone. We are still waiting for people to connect, so we're gonna wait for a couple of few minutes more, just to give chance to everyone who wants to attend to connect now, and then we get this started. So be patient and keep with us for another couple of minutes. Repeating for those that are just connecting now, we're waiting for a few minutes to have everyone connected. So, yeah, we will be starting in a couple of minutes. Okay, as numbers are accruing, I think we can get this started. So welcome you all to the single cell and spatial metabolomics online day of 2025. I'm very happy to introduce this event, which I have to say has been organized basically only solely and completely by PhD students from the labs of Joele Lamanno, Theodore Alexandrov, and my lab. So I would like to thank the sponsors of these events, which are the EPFL, the university from where this is broadcasted, the Human Cell Atlas, and the Sphingolipid Biology. And I would like actually to name the people that have really that have really contributed to make this event what it is now and for the second year in a row. And so first of all, I would like to thank Luca Fusar-Bassini has been really the engine behind this entire thing. He is now following us from Honolulu. So thank you, Luca, for being here, though, with a big time gap in between. And then Nika Gorshek, Alessandro Valente, Xinjin Ang, and Jai and Lin, who are actually taking care of the broadcasting today. So these are the people that you will see mostly moderating the Q&A and other, and introducing the speakers. And then also the other people that have been involved, like Ali Reza and Bishoy from Theodore Alexandrov Lab. They are the people that really made us. So we are actually here to take advantage of this gigantic journal club that we will have today. And I would like to say that, you know, as a general kind of introduction, I think this is very timely. This is an event that really comes in a moment where basically we are transitioning from using technology to, you know, to try to understand what is possible to be done, to using the same technologies now to really understand biology. So this is the time to have fun with what we actually are able to do. And, you know, and a couple of things I want to say about how the event is going to be run. You will see we have speakers that are going to be, you know, giving their talks one after the other. At the end of each talk, we will have time for a few questions. And in order to ask questions, please type them in the Q&A chat that you find in your Zoom. The questions will be read and selected, but just by, you know, in a first come, first serve kind of fashion by the moderators, and will be apparent to everyone. They will be visible to everyone the moment that they've been answered. So some will be picked up for, you know, oral answer, and some will be kept in the chat to be answered after by the speaker when the time is right, let's say. And with this, I don't want to steal any more time to this event, so I will actually pass the microphone and the virtual microphone to the organizer. In this case, I think it's going to be either Alessandro or others to introduce our first speaker, that is Martin Guerra. I don't know whether Martin wants to start right away, but thank you so much for being here, and thank you, Martin. I stopped sharing my screen, and it's your time to start your talk. Thank you so much.
Hello, everybody. So I'm Alessandro Valente. We are here. The first speaker is Martin Guerra, that I see already online. So please, Martin, the stage is yours.
I have to share my whole screen. I hope that works. Let's see. You please tell me, because I don't see you guys anymore. You see it?
Perfect like it is. So please
Okay,
go.
excellent. So thanks a lot for inviting me. This is quite a nice lineup of speakers. I'm really excited. And yeah, thanks again. Yeah, okay, let's kick it off with lipidomics, metabolomics going spatial. I mean, maybe for a better title, I chose this one. But before I show you some of our work we're doing in this arena of spatial single-cell lipidomics, metabolomics, I have to convince myself that it's worthwhile to do single-cell and spatial lipidomics and metabolomics. Frankly, I have always been very critical about it. I still am very critical about it, which I think is a good thing for a scientist, because, you know, you can generate a lot of nice, fancy pictures and photos and whatnot, but what's the added value if you compare it to a bar graph? And when the field kicked off, frankly, not a lot of people could give me a convincing argument why it would be better than a bar graph. However, things have evolved, and I'm getting more and more involved in it. And one of the things I think comes a little bit from this review I wrote with Gary Sustek a while ago, where we try to more strongly integrate the metabolome into the central dogma of biology, which runs from DNA to RNA, proteins, and metabolites. And what we didn't do back then, but I think we realized this nowadays, is that next to these molecular levels, there's also the cellular level. And different cell types communicate by exchanging metabolites, proteins, and signal molecules with each other. And in this, you know, conceptually in this field, it makes a lot of sense to then look at cells in their natural surrounding, in their natural habit. Now, my journey to finally accept that single cell and spatial metabolomics and lipidomics is worthwhile started with the work of Chris Glass, which we will see in a little bit. The other realization, I think, and also this I reviewed together with Gary a while ago for cell metabolism, which I think quite nicely shows you that, you know, throughout the history of biochemistry, biochemical advance has always been going hand in hand with technological advances. And I mean, this is quite clearly shown also for the field of spatial and single cell technologies. I mean, mass spectrometers became more and more sensitive, so that single cell was in reach at all. Spatial technologies developed, and I think we are not at the end. This will continue for a while, but also here, you know, 20 years ago, a lot of things were not possible that are possible now. And I think, yeah, we should use these things. But I will also show you a couple of examples where we should be careful and maybe not block ourselves, but critically think about the results that we obtain from these technologies. Now, so as I already told you, my personal journey into this started a little bit with my work on targeting the enzyme dehydrocholesterol reductase 24, which we're working on at the therapeutic modality. We see some brief results there, and this kicked off with actually with Chris Glass's work in Cell from 2012, where he analyzed rather cell-specifically foam cells. And what he found is that foam cells, that they are intrinsically not pro-inflammatory, what everyone expected, but they have a counter mechanism down-regulating the expression of DHCR24 and thereby accumulating a cholesterol precursor called desmosterol. Now, what he didn't know is that I had made inhibitors for exactly this enzyme in my PhD, and frankly, we didn't really know what to do with them. But when I saw the report by Chris, we took them out of the closet again, and we optimized them, and we kicked off, let's say, a drug development research line in the lab based on this enzyme. So it basically goes like this. One of our published inhibitor is this one. It blocks the enzyme DHCR24. This will increase the cholesterol precursor desmosterol, which is an endogenous liver X receptor agonist with pronounced anti-inflammatory effects without co-activating the sterol response element binding proteins, which is the predominant side effect of classic liver X receptor agonists. Now, a little later, Chris also already kind of hinted that this desmosterol seems to have cell-specific effects. Now we're again in the field of, you know, spatial, single cell, cell-specific analysis in whole organs. And in a personal communication with Chris, he told me he had quite a hard time of publishing this because no one really wanted to believe him in the first instance that this desmosterol LXR mechanism is predominantly triggered. He published it for macrophages, but now our work over the last years shows predominantly in phagocytes. Now, so we stumbled across the exact same mechanism also some years ago again, and this time in the context of glioblastoma, where we investigated tumor-associated macrophages, and specifically lipid-laden macrophages, together with a group of Laila Akari from the Dutch Cancer Institute. And you can read up on the details in the Cell paper, but this was the exact same molecular mechanism that is very cell-specifically pronounced in these lipid-laden phagocytes. Now, a little bit in parallel, colleagues from the Max Planck Institute in Göttingen, Gesine Saar, Stefan Berghoff, and Lena Schmid, they pointed to also this mechanism in microglia. So again, we're talking phagocytes. Now, I've met Lena, Stefan, and Gesine on a conference. Said, well, why don't you guys test our inhibitors we made for this DHCR24 enzyme, and we'll see, well, this does anything in multiple sclerosis. And so they did, and it worked. But then, you know, there comes the question, yeah, so how does this work? Is there now really a cell-specific effect? Is it really for a specific cell? How can we show that it only works in microglia, that it doesn't affect astrocytes? And then this, you know, in the setting of a whole brain, I think now we're talking spatial lipidomics and metabolomics. Now, to approximate this, we started with a tritium-labeled probe first thing to show maybe now that you can target a certain organ before you aim higher to a specific cell. We made tritium labeled probe, and as you can see here, the inhibitor goes into the brain. That has just recently been published in Immunity in a collaboration with a couple of groups, and especially Mika Simon's lab from Munich. So inhibitor goes into the brain and can does its magic there, because we didn't know exactly about a mode of action. Now, to conclude this, why is single cell and spatial analysis important? By combining spatial transcriptomics, stainings, as you see here, for example, for GPNMB or PLIN2, conditional knockouts as this ABCA1G1 microglia-specific knockout that the colleagues in Munich made, showing that indeed this signaling path from DHCR24, desmosterol, liver X receptor, ABCA1 cassette transporter upregulation is involved in the disease, at least in a mouse model of multiple sclerosis, significantly enhances disease severity if you have a microglia-specific knockout. And you can accompany this by some stainings, etc., etc. We don't have to go into the details. Also here, you can read up on this in the publication. Here again, in parallel, colleagues from Leiden University, Mario van der Stel's group, they did more of a lipidomics investigation of such multiple sclerosis lesions in humans. We did it with spatial transcriptomics, in this case with Murfish. But to cut a long story short, basically the spatial analysis and the single cell or the cell, let's call it the cell-specific analysis, ultimately showed us what the exact mode of action of our inhibitors is and in which type of cell type they work. So this kind of convinced me personally that it's worthwhile to dig into this further and to expand activities there. And the new kid on the block is, I guess, still the mass spectrometry imaging part, the part which is, used for mapping the lipidome and the metabolome. Spatial transcriptomics platforms are much more integrated nowadays. So let's have a little bit of a look what we're doing there, and also at some caveats and raise some caveats and some pitfalls in this arena. So to spatially map metabolites and lipids, most people like my lab, we reside on MALDI mass spectrometry imaging, but there's of course also DESI, but we will only focus on MALDI. Just very briefly, how does MALDI MSI or mass spectrometry imaging in general work? I think most of you know this. You have a tissue slide, you spray it with a matrix, and then you have a laser basically going over it in XY coordinates and collecting all the data from which you then can generate the molecular images. Now, normally it goes like this: tissue section, matrix spraying, and then the MALDI MSI analysis, as I told you. But this gives already, and this is well known, quite some analytical pitfalls, even of intra-tissue comparability, but even stronger inter-tissue comparability. How quantitative are your results? And annotating the observed molecular features is non-trivial. Now, to overcome this, people have started a while ago to introduce internal standards. I think Shane Ellis and Ron Heere were some of the first to introduce the internal standard concept for lipid classes. So to use one lipid class specific internal standard and use this to correct, and that worked quite nicely. Now, the field of metabolomics, unfortunately, is a little bit more tricky. What works nicely for lipids, these lipid class specific internal standards do not always do the trick for metabolites, because metabolites are chemically more structurally diverse, and especially their mass spectrometric fragmentation doesn't follow the same nice rules as you can sketch them for the lipids. So that's why we came up with the idea, well, why not use 13C labeled yeast extract? That comes from the very old work from the 90s, actually also from the Netherlands, from Walter van Gulik's group in Delft, who were the first to come up with the idea, well, why don't we completely label yeast? And then we have this beautiful, fully labeled extract material, and we can basically make pairs of all the metabolites we detect and correct each metabolite for its authentic matching internal standard. And so we did. And what you see here is a comparison between the classic root to mean square or total ion count normalizations with an internal standard normalization for each and every single metabolite, the matching internal standard from a 13C labeled yeast extract. And as you will see down here, is for example, for palmitate, you can get quite some diverse images. So if you do not correct, for example, the palmitate for its internal standard, you would think that it accumulates in the cortex of this kidney, while actually it's much more in the middle and much more finely distributed rather than in big chunks like you would see here. And this also holds true for root to mean square. So I think this is a step forward, especially the internal standard work also introduced by Shane and Ron. I think it's a very important analytical measure to have more reliable images and more meaningful images in the future. But so we dig further a little bit into about that, does it then have any kind of physiological relevance? I mean, maybe you just get the same result in a disease model when you don't do any normalization. So we did use the photothrombotic lesion model in a stroke model, where you basically have the two hemispheres. One is control, one is diseased, so to say, and we carried out the workflow. And also here, long story short, it's in Banget Alie in Nature Metabolism. What we could find was actually an ipsilateral region that is not within the stroke area here, but cluster 16 here, which the guys from the stroke group were very surprised about, because their myelin basic stain and their DAPI stains didn't show any major differences in the cluster. However, using our advanced MSI approach, which we doped QMSI, actually made us realize that this whole region is metabolically reshaped even 28 days after the insult. However, it's not to be seen on MVP or a DAPI stain, and there was various metabolites and lipids actually being changed in this region. We took it even one step further. We said, okay, let's just try it. We make some calibration lines. You can buy this yeast extract labeled and unlabeled. We quantify the whole thing with NMR. So we have a ground truth analysis. And then again, we compare root to mean square tick, NMR, etc., etc. And frankly, I mean, this looked better than I would have expected. I really thought this would be all over the place, but it looked quite decent, I have to say. So I think it's not a solution. It's not a silver bullet, right? Overcoming all kinds of problems we have, but at least it's a step into the right direction. Now, let's use the same example and dig a little bit into annotations in the last couple of minutes I have. So another molecular species we found was accumulating in the rim of the stroke, which was very interesting. And it was basically 865.50. And these are different mice. So you see it's really reproducible. And then, what you normally do, you're right, you go to lipid maps. We knew it should be sort of a lipid. You have a couple of hits, but then normally you're stuck. And from a biological perspective, if you look at the two top hits, I mean, I don't like this LBPA nomenclature, but that's a different topic. It would be BMP, bismonoacylphosphate, the lysosomal lipid 4412, or phosphatidyliglycerol. And biologically, I mean, these are, there's a significant difference in the function and in the occurrence of these two lipids. So it's very important to have this correct. And it took me a while until one evening or one night I woke up in the lab and I thought, well, I'm kind of stupid. This has to be a BMP lipid, because it basically pinpoints to the phagocytes, upregulating the lysosome. And then what we see is lysosomal lipids. And this then, is like a positive example. So with the new technologies coming up, like in this case, the TIMS domain, in the CCS domain, so in the ion mobility, we could then separate the PG and the BMP and really clearly pinpoint, like, look, guys, what we mainly see here is really a BMP lipid pinpointing to lysosomal activity in this area, which is kind of logic. I mean, but still, you can now prove it. And I think like this, with the PASEF and the other developments, I think there's a lot of nice developments now coming into the field, and I see people using them. So it's great to see this, and also great to see that the annotations are getting better. And, you know, don't get me wrong, everyone is allowed to make mistakes. We make mistakes, everyone makes mistakes. So it's just of trying to improve it. And that brings me to the last part, something that I think we overlook still a little bit, and we all are aware there's a loss of phosphatidic acids in MALDI images. It's most of it, and I think Theodore also had a paper about it. Most of it is in-source fragmentation. I had this little piece, it's only one page, again, together with Gary, where we looked into Medlin and 950,000 molecules, and we found a significant amount of in-source fragments in electrospray ionization. We got a lot of pushback on this, and there was a recent paper also outlining that it might not be biologically important, etc., etc. So, you know, again, here we looked a little bit closer into this. And let's circle back to the example I told you before, the DHCR24 and the SH42. So I told you we used it in multiple sclerosis. We wanted to know in which exact cell type this is. Actually, this was accepted this morning provisionally in analytical chemistry. So you can also soon look this up. So desmosterol bumps is increased when we give SH42. So we used Roberto Angelini's derivatization approach. I mean, it really couldn't get below 10 micron. I mean, more or less 20 micron. But, you know, if you do GCMS on bulk and you do the Roberto's derivatization approach, you kind of see the same result. You bump up desmosterol when you give the inhibitor. But we couldn't get to single cell because we wanted to show, okay, is this now selectively in microglia, for example? So we said, okay, MALDI-2, that's the solution, right? I mean, now we can go to 5 micron. We have the machine, we have the microgrid, whatnot. We tested all kinds of matrices, and we didn't see any difference anymore. So, you know, we started to look into this, and we tested all kinds of matrices. But ultimately, in the end, what did bite us in the ass is in-source fragmentation in the MALDI-2 source. So basically what happens is that you get a dehydrogenation product from cholesterol, and that forms the exact same mass as desmosterol, basically obscuring the accumulation of desmosterol. So saying that in-source fragmentation is not biologically relevant, I'm not so sure about it, because in this case, this would have sent us into a rabbit hole, but we would have thought, well, SH42 doesn't work in the brain. If we would not have had our tritium results in our GCMS, this would have made me conclude, okay, there's no difference. It's not significantly increasing desmosterol in the brain. So we leave it there, you know, something else is happening. Before I conclude, so where is then, you know, I already told you, you know, quantitation, in-source fragments, more targeted approaches. I think it's all new fields that are developing in the MSI arena. And I think the other one is then, you know, carbon tracing, carbon flux, the traffic jam paradox. If you take a photo of running traffic, you think it's a traffic jam while it isn't. We've done some work here also. One example would be regulatory T cells, conventional T cells, and they have very specific metabolic demands in terms of what they like to eat. And also, you know, if you think about this from, let's say, a Darwinistic point of view, this could also, you know, funnel into why certain cell types proliferate or work in a certain surrounding. I mean, if you try to raise a cat as a vegetarian, you will likely not proceed. We've done a couple of work in this arena. It's non-trivial, and it only works for a couple of metabolites at this stage. I think we had some 10 or 15, and it's a lot of computational work, which brings me to the last point. And Paola, my PhD student, will tell you a little bit more about this. The other, I think, big elephant in the room, where a lot of improvements I think are necessary, is to cope with all these data we are gathering. It's so much, and it sometimes takes so long to upload anything into Skills. Can already take you a day before you can even look at it. So Paola will tell you some of our activities in this field, where she came up together with Yassine from my group, with I think quite an innovative new angle on how to do this a lot faster. With this, that's Paola. We will see you later. Thanks to my group, and yeah, again, thanks for inviting me and giving me this chance for showing some of our work. I'm happy to take some questions.
Okay, I think Alessandro, are you gonna run this one, or Ali Reza?
Yeah. Yeah, can you hear me now?
Yes.
Yeah, thank you, Professor Guerra. Very nice talk. Is there any question right now from anyone? If someone wants, we can give them access. There's a hand
There
here.
are two people that actually raised their hands. I invite them to write the question in the Q&A, and three people actually in the Q&A, and this will be read by you, Ali Reza. Otherwise, this is gonna become very difficult to deal with.
But still, I haven't received any.
Yeah, I see this, but
Yeah.
they've raised their hands, so please. But there is one from Joelle that you can read in the.
Yeah.
Okay.
So Joella has asked, is there some computational heuristic, maybe using machine learning, that we can learn to rescue experiments performed without these important internal controls? In other words, there is a chance, is there any chance that we can learn a correcting function with machine learning or otherwise?
Yeah, very good question. The answer is I don't know, but I think it points into a good direction. I mean, first of all, as I said earlier, I think we should use it for what it is. Basically, what I wanted to say is just be critical about the results you obtain, and frame them in the right way. Don't just, I mean, I've seen so many people, they just take a peak list and they put it into lipid maps and they say, okay, look, there's this lipid. This is not how it works. And this is not what you want if you want to sketch biology in a spatial fashion. So again, I think it's good what we have, and there's a lot of cool stuff coming up. I mean, you guys work on the 3D brain lipidome was really awesome, I think. And more is developing, but I think, yeah, again, there's still a lot to be careful with and a lot to do. But how to correct it for this, I think we still need more knowledge about what is actually really happening in MALDI and MALDI-2, and what kind of in-source fragments do we get from the different chemical entities. So there is not a lot of knowledge what would lead to what. If you know that, then potentially you could design a machine learning algorithm to correct, yes.
Amazing. Thanks again for your talk. I mean, there are still like two other questions in the chat, in the Q&A, but unfortunately we are out of time. So if it's possible for you to answer them in the chat,
I will do so. Yes.
Thank you so much. So we thank again Professor Guerra for his amazing talk, and invite the next speaker, who is Joella Lamano from EPFL. Joella, the stage is yours.
Hello, everyone. Can you see my screen in the right, in the presenter mode, in the right mode? Fantastic.
Yes.
Wonderful. It's fantastic to have the chance to tell you this story today. I was also very excited by Martin's talk, and definitely many of the indication really resonate with me. So the story I'm going to tell you about today is at the intersection between neuroscience and metabolomics, and is very much in line, I think, with the ambition in neuroscience to characterize the cellular and molecular diversity of the brain. And you probably know that this is initiated a century ago with people like Hal, but nowadays, fast forwarding to the present, we in the nervous system, we have basically a model of cellular heterogeneity, which extends to the level of really thousands of transcriptional defined cell types. And typically, people talk about 5,000 cell states in the brain. So The transcriptome is not, of course, the all part of the story, and molecular heterogeneity goes further. And today I'm here to talk about actually a very important axis, the lipidome. And even before showing any data from the literature, we can clearly tell that lipids have a central role in the brain. We know that they influence action potential. We know about myelination and Rambiers nodes. We know about their influence on signaling response, neuron activity through the synapses, structural organization, and transmission. And I think they're also a pretty good proxy for overall metabolic state. So, long story short, and going straight to the point, together with the D'Angelo lab, we have recently launched an effort to map the adult mouse brain lipidome. This is basically was done by collecting multi-imaging data at 25 micron resolution. So we have desorption spots at that are 5 microns distance by 20 micron. And we did this across 11 brains, spanning different spanning the sexes and also pregnancy. We annotated 172 lipids with high confidence and analyzed in total 7.2 million pixels. So when we were doing this and when we were launching this effort, we know actually already that MSI data processing has limitations. We already heard part of this from Martin, and we also, some difficulties are data driven, are at the level of really data processing. But one cool thing is that we also knew that we could overcome part of this using our tool Umaya. This is a recent work that was recently published in Nature Methods. And basically what this is an end-to-end computational framework that redesigns basically several standard steps with one goal in mind, which is try to carry over as much as possible of the data to the final step, or try to homogenize it so that you can build atlases. And then retrospectively, you can also, of course, do all the filtering you want and, QC, but try sort of to really homogenize as much as you can from this spectra. The results that come from this approach of preprocessing the data are shown here. So you see in every row a set of our samples across the anterior posterior axis of the mouse adult brain. Each row is colored by 3 lipids. You see the specific species. You can appreciate not only the really strong spatial patterns observed, but also that some species of the same class actually have quite different patterns. So that's where we started from, and we, of course, also important part of the resolution that we have is in 3D. So with over 100 sections, we really have a very good anterior posterior resolution, and we can really interpolate in sort of a 3D fashion, getting information about different lipids across the entire axis. So the first step in an analysis of a dataset like this is having a good reference model. And typically this is done by the omics field by clustering the data. Clustering, again, needs to be seen as kind of a model of the variability of the data. It's a discrete model, right? One could imagine different models, maybe a more continuous one, but clustering is a very good and pragmatic one. We proceed in the following fashion. We first do a low-dimensional we try to do a dimensionality reduction using non-negative matrix factorization. We think this is particularly informative and also reduces a bit the noise. It allows us to launch basically a iterative binary splitting clustering approach. So we basically iteratively split populations in a feature space that is being selected by variable lipids. And we not only limit ourselves to do the split, but we check it in different ways. We train a classifier to check separability with a train test step split. And this is very cool because, having a teach split, this validation also allows, post hoc to break the instance dependence of clustering so that we can repeat the same approach, not a clustering anymore, but sort of classifying. So any single section that we can give then now to this engine will again reproduce the clustering and this same clustering stratification model that we can produce. So we've been doing this. We have some set of stopping criteria, in particular based on this kind of test validation set scheme. And what we get out of this is a classification of lipid heterogeneity in the brain in 539 lipid zones. So similarly to what the Allen Institute does for transcriptional heterogeneity, what we do here is that we also organize this, and this comes natural from the clustering, in a hierarchy. So down at the bottom, you have the single lipid zones, here you see their abundance, as well as the representation that they have across different Allen main divisions, anatomical main divisions. So you can appreciate already of some lipids really anatomical, and they really have quite a strong pattern in terms of their localization. The other things you see can see other level of hierarchy. So, for example, you see classes, and even sort of the broad level 1 classes. And I think this is actually important. I want to stress this, because really what we are seeing analyzing our data is that I would totally feel like recommending strongly the community to seriously consider at least these axes of heterogeneity when studying the lipidome of the brain. We really see in our data a high risk for confounding the results of everything we do if sort of at least this level of heterogeneity is not acknowledged. So we would really actually advocate for stratifying the results in lipidomics based at least on this higher level of classification. So the results of the clusterings look a bit like this. This representation, is not in scale. Each dot here is actually smaller, it's 5 micron, and the dots are spaced by 20 microns. And this is the clustering overlaid to a zoom in, for example, of the hippocampus. It gives you an idea of the granularity we have. This scattering, the local variability that you see here, is not noise. Nearby pixels often differ because actually represent different cells that are sampled, right? And here with 5 micron, we typically samples, just a couple of cells or hopefully one cell. And, this, I'm going to show you later that we have all convincing evidence that this is really not noise, but actually correlates a lot with biology. Just to get a bit of a visualization that, there are not enough colors to see these lipid zones. Though they're appearing in front of your eyes one by one. You get maybe a bit of an appreciation of how they are localized and they're fine-grained. Also more broadly, you can have a bit of an outlook of how they look across all the sections we collected. And you see how uniform across regions and anatomical they are, right? And this kind of gives us further confidence we're really hitting and kind of catching something that of relevance. So yes, so these lipid zones are anatomically organized. So this maybe starts to ring a bell with things like cell type. But before going there, we started to ask a bit more kind of a general question, a bit conceptual information content question. Given a pixel and its representation in lipid space, is this enough information to act as a zip code for the address of that vector? In other words, is the lipidome a zip code for a position in the brain? And we try to do this in a most naive way, sort of, most farther from overfitting you can imagine. We take a vanilla MLP, no metaparameter Turing, and we try to predict from the vector lipid expression the localization in space. And this is like the predictions, across learning. So we learn, and that's, you see now a brain is forming. So every pixel can be, every lipid composition can be used to predict rather well the original localization from where that pixel was sampled from. There is a lot of spatial information, and of course this makes us think, the first hypothesis, that there might be a connection between lipid heterogeneity and underlying cell type granularity of the brain. So indeed, if one looks carefully, and this is why I'm showing you the zip map, at the finest level of lipid zones, we start, and want to stress at the finer levels of our cluster, and we start to capture cell types. And I'm showing here basically the normalized colocalization score between the lipid zone and some cell types. And just to get a bit of a visual catch of what we're catching, visual idea of what we're catching here is, in the left, here in red, you have some example of some lipid zones, and on the right, the most spatially correlated Allen cell type. This is like fine cell type that has been defined by the Allen Institute independently in two sections that are independent, they're registered into each other without, of course, seeing the clustering beforehand. So, well, then there is this strong association with cell type, but does it mean then that the transcriptome predict the lipidomes? It's just, so simple. The answer is no and a bit. It depends what you're really asking. So if the question is whether we can do it in a principled way from the associated enzyme, like can we take basically every enzyme and sort of the reaction that catalyzes and looking at the lipid and see where it correlates, we see the answer is definitely not. We cannot do this prediction. It's all over the place. It ranges from very good correlation, very bad, even within the same class and across different species of the same class. So this can't be done. What can be done instead, and that's why I said a bit, is to try to use the sort of supervised learning approach, where we try to use any kind of gene expression we have access to with kind of large-scale Murphy dataset, and we try to obtain a decent reconstruction of the lipidome. And the results are not bad, right? We can get something that almost 0.7 Pearson correlation, right? So yeah, but here we're using much wider gene set than even metabolic genes, and that's what I'm showing here. So there, we are considering different gene sets across different level of the lipid zone hierarchy. And so here we are at the very first early splits, and towards we go to the right, we have like fine cell types. And what you see even that the categories of genes that predict the split change while we get to fine grain level. We basically go from in the middle, sort of from genes that relate to membrane organization towards genes that are involved in neural cell biology and fine grain cell type identity. We're basically getting to a same sort of similar latent space. That's why we can predict. But of course, this is not an outgroup prediction. We are, of course, still predicting within an healthy brain. Another important angle I want to stress and emerge by looking at the hierarchy. What I'm showing here is the different level of the hierarchy, and we're going down of the lipidome. And what I'm showing in these dots is how cell type heterogeneity follows. So this is the corresponding cell types that exist at each split according to the Allen classification. And sort of cell types basically spread across lipid zone in ways that do not align with transcriptomic hierarchies. This is not contradictory with what I said before, because when you get to the fine grain level, then it starts to match once again, because you're accessing the fine grain level of the possibility of each cell type to regulate metabolism, right? But truly the information at the global level, at any hierarchical level, is actually pretty different. Another unexpected angle we found out in our study is that since we measure lipidomes and we're not limited to segmented data and to somata, we can actually see and we start to capture dendritic and axonal compartments. So we basically see patterns related to connectivity. What I'm showing in this a bit kind of complicated visualization is a Cree driver line that from the Allen mesoconnectome that highlights in with colors from red to blue a stream of a particular neural type from start to end. And we find in many cases, over 70, that it overlaps very well with one lipid zone. So we see basically different lipid zone co-localizing with the entire stream, the source and the target, axonal and dendritic arborization. So something we not expect to see through the lipidome, actually a bit of the connectome. So other more kind of neuroscience focus finding. One thing that we observed that we were also surprised as when we look at the white matter is the white matter heterogeneity. Typically, transcriptomic studies defined up to maybe 10 types of oligodendrocytes in the white matter, but we detect here 65 distinct white matter related lipid zones and fiber track lipid zones. So this basically show an anatomical specificity that is not seen in transcriptomics, and we think those clusters are very credible because they are anatomically, they may also make a lot of sense. Another reason that another fact that we found about these lipid zones is that they actually split in sort of a broad set of categories, two categories that have very different mediolateral distribution. And basically forming a gradient that is mediolateral that has also been reported in epigenomic studies where they link this chronotopic pattern to developmental timing and regional biology. So to conclude, I want to ask, we wanted to ask another question. I mean, we did much more in the study. I recommend you to read it if you're interested in the metabolism of the brain. So we want to ask, can the lipidome, and in particular the lipid zone model, serve any use in practical use in biology? And in particular, we're interested, can we study in any way physiological adaptation using this model? And one convincing example comes from a physiological perturbation, in a sense, that is pregnancy, that we also profiled, and we try to interpret using our atlas. And what we can appreciate from this heat map that summarizes all the results, showing in the columns the different lipids and the rows the different lipid zone supertypes, is that the response to sort of the adaptation to pregnancy, they are not global. They're very specific group of lipid classes acts on very specific lipid zone classes. So in this level of granularity is basically lost if you start to stratify just by regions, right? And so it's very important to use this model to be able to detect these adaptations. I wanted just to show two examples and conclude. One where we actually found a lipid zone in the cortex, in the outer layer of the cortex, that really kind of significantly expand, and that we're currently validating. And another case where we really see an increase of, class of lipids, sphingolipids in particular, XR42-2 in the white matter. And with this, I really conclude, just redirect you to the paper and our online resource that you can use to actually ask your question and query your lipids. I think we try to make it as much as possible user-friendly, and just wanted to sort of summarize our conclusion. So lipid zones are biochemical territories, are a model, right, that aligns with cell type, but reflects a different logic. That by studying lipid zone, we can reveal signature also of connectivity and development. It's quite interesting, something to look and sort of delve deeper into. White matter and oligodendrocytes show an unexpected biochemical diversity, really much farther from what we were expecting. And that pregnancy induces a structured lipid zone specific shift. That I think this demonstrates that lipid zones are a sensitive model for physiological adaptation study. And with this, I want to thank all the people involved. First and foremost, Giovanni D'Angelo, and from which we started, just me and him, this project, and then of course been taken the lead by Luca. That's been really incredible in the analysis of this, and all the people collecting the data and supporting this effort. Thank you very much.
Thanks, Joelle, for the fantastic talk. Now, there is no time for questions, unfortunately, so maybe I will ask Andre Nadler, which is our next speaker, to step in.
Can you hear me? Wonderful.
Andre, can you hear us?
I can hear you. And start sharing and start my presentation. And I hope that you see the correct screen.
Yep.
Everything good? Wonderful. Yeah, so many thanks for inviting me, and I really appreciate the opportunity to speak here. And we will continue on the topic of lipid imaging, but we will go down in scale by a couple order of magnitudes, and I will talk about imaging lipids on the scale of organelles and below. And why is that important? If you think about lipids, it becomes clear that all free living organisms through all three kingdoms of life share relatively few very conserved things. No? So there's, of course, among them the genetic code, ribosomal protein biosynthesis, but also lipid membranes. And these shared elements are the most fundamental features of cellular life. And the primary function of lipid membranes is to compartmentalize lipid biochemistry. The more complex a reaction, the more complex a cell becomes, or an organism becomes, the more specialized reaction chambers it needs, and that's reflected in the evolution of the lipidome. And the mammalian cell contains about 2,000 molecular distinct lipid species, and this number continues to go up. And I don't have to tell this audience that we really do not know what most of these molecules do. Well, one reason why that is that we do not see them. And that is one motivation for starting to image lipids. The other one is that lipids are at the heart of global health challenges. Many metabolic or neurodegenerative diseases are either bona fide lipid dysregulations, if you think about metabolic syndrome, non-alcoholic fatty liver disease, or cardiovascular disease, or have a strong lipid dysregulation component. And a typical example is Alzheimer's. And the fundamental importance of lipids for both basic biology, but also for translational research means that they should be like a huge and thriving field of lipid cell biology. And I think Giovanni D'Angelo will like that. But it's only been the last couple of years where progress really has accelerated. And one of the key aspects is really that technology for imaging lipids, both at the cell and the tissue level, is just coming online with a quality that we can really address mechanistic questions. And there's many open questions in lipid biology that remain unanswered. But the one that I'm most interested in is essentially why cells maintain or host cells maintain organelle lipid compositions. I think I have started the wrong presentation. And I go back to the... yeah. That's the one. Okay. That was the one from Paris last month. This is the one for today. It's slightly shorter. Sorry, guys. And the question is why such fundamental questions are still unanswered. And one key aspect is that there are fundamental differences in that make it difficult to really trace lipids in the cell. One of them is the so-called lipid diversity. And that means that the differences between individual lipid species are typically tiny. Also, you either have big differences in linkage, those are, for instance, these ether and ester linkages. You have different numbers of double bonds, you have different chain length. All of them have in common that they are a few atoms at most. And that means you can't really stick huge marker groups on lipids and then expect these probes to behave in the same way as the parent molecules do. Also, you can only use minimal modifications for tracing lipids in cells, or you have to have some way of monitoring the native species, which is difficult. And the second thing is, and I think this also shown through in the prediction of the lipidome from transcriptomic data, is that the lipid structure and localization is not fully genetically encoded. And what do I mean by that? The structure of individual lipids is the outflow of a complex network of lipid metabolic processes. And there's not, in a similar way, a function where a certain sequence of nucleic acids is directly causing a certain sequence in a protein structure. And there's a second layer of control on lipid localization, and that are the lipid transport pathways, where you have vesicular modes of transport, where vesicles are exchanged between individual membranes, but also non-vesicular transport of lipids via lipid transfer proteins at membrane contact sites and through cytoplasm between individual organelle membranes. And taken together, that means that genetic perturbation approaches are limited. Well, if you perturb somewhere within these networks, you will always get a pleiotropic effect. There will not be the single lipid that is changed in abundance or localization. You will always have a different outcome on the level of the lipidome. That means that the typical cell biological approach of either doing a genetic perturbation or following the localization of a GFP fusion protein really doesn't work as well for lipids. And that is why progress in lipid biology has been slower. Also, we can't use the well-established discovery pipeline of cell biology that has been working so well for proteins. What we are using are so-called bifunctional lipids. So we introduce a diazurin group that we can use for UV-mediated cross-linking to other biomolecules, but also to trace the metabolism of these probes by mass spectrometry, as this is akin to an isotope label. Also, it gives you a distinct mass that you can follow as these probes getting metabolized. And then we have an alkyne here at the tip, and that we can use to attach reporter groups, Roforce, but also biotin for pull down. We are not the first ones to work with these probes. There's many groups using them, but I think we are the first group that really has put this together into a lipid cell biology workflow based on minimally modified chemical probes. Now, what can you do with that? You can use it for direct visualization of lipid localization using nanogold labeling in EM, and I'm showing you here sphingomyelin molecules on the plasma membrane of a cell labeled with nanogold. You can use it for correlative light and electron microscopy of lipids and proteins. So here I'm showing you an example of differential trafficking of sphingomyelin transferrin, the endosomal system. You can use STEP imaging of lipids to look at partitioning of individual lipids into, for instance, clathrin-coated pits and other endosomal structures. And you can trace lipid-protein interactomes by mass spectrometry. Okay, so what do we do to make all of this possible? And here I'd like to give credit to Alf Honigmann. So basically the first lipid imaging data that were really functional, we developed together back in the day when we were still working in the lab. And this is really the team that put it all in motion and acquired all the data sets that we recently published. So we make these bifunctional lipids in the chemistry lab. We load them into the plasma membrane of living cells using an alpha-methyl cyclodextrin-mediated exchange reaction. And by now, we can also reconstitute uptake of lipoprotein particles that we load with these bifunctional lipids. We do classical pulse chase experiments to allow for transport to endomembranes, followed by UV cross-linking, self-fixation permeabilization, fluorescent labeling, and then imaging and image analysis. Similarly, we can skip the UV cross-linking step, do a lipid extraction, and then follow metabolism. And finally, we can skip the self-fixation step, do a protein pull down, and then look at lipid-protein interactomes. To map lipid flux through the cell, and by lipid flux, I mean the movement of lipid components both through the physical space of the cell, so that's transport, and the chemical space of the cell, and that is metabolism. We made quite a set of these probes, and this has been expanded in the meantime, where we have, for instance, sphingomyelin phosphatidic acid as one of the key intermediates in lipid metabolism, phosphatidylethanolamine, and an entire series of PC species that allow us to track the influence of side chain composition with regard to both unsaturation degrees. So we have polyunsaturated species, saturated species, monounsaturated species, but also positioning, SN1, SN2. So these three guys and these three guys are regioisomers. And this allows us to trace lipid transport through the cell. Also, I'm comparing here two PC species, one that is fully saturated, one that's polyunsaturated. And I'm showing the localization of this lipid at 4 minutes. What you see is that the saturated species still is primarily at the plasma membrane. You see a couple of bright dots. These are endosomes. And then if you really squint, you can see the nuclear envelope here. Also, a little bit of that material has reached the ER. However, the polyunsaturated species is already almost completely internalized after 4 minutes. So there's a major difference in transport, and that is reflected in the later time points, where at half an hour, you still see quite a bit of plasma membrane signal for the saturated species, which is completely absent in the polyunsaturated species. And at half an hour, at 4 hours, you basically reach steady state for both of them. Now, these are relatively complex images, and we can go into a bit more detail of how we analyze that. Also, I have here essentially the equivalent of POPC in its bifunctional version. And at half an hour, much of that is at the plasma membrane. Quite a bit is in the ER. You see bright structures here, which are presumably Golgi, and then these even brighter structures, which are endosomes. And while by eye, it's easy to look at this image and see what it can be, it's difficult to get hard numbers out of these complex fluorescence microscopy images. And what we do is that we do counter stains with a number of organelle markers. In this case, we have the plasma membrane, the ER, and the Golgi. And then we train pixel classifiers that essentially learn how these organelles look like. And this allows us to segment these organelle markers, not the lipid imaging channel, these organelle markers, and generate probability masks for that tell us for each pixel to which extent this belongs to each one organelle in the composite image. And we use the regions of the organelle marker masks that have no overlap with each other to derive the pixel distribution in the lipid channel. And these pixel distributions we then use to assign signal that is in overlapping regions to each one organelle. And this is how it looks like for this particular image. So this is the signal we assign to the plasma membrane. This is the signal we assign to the ER, and this is the signal we assign to the Golgi apparatus. And then we can quantify simply by calculating the fraction of the overall integrated fluorescence intensity between these different organelles. And now we did this for the entire set of lipid probes that I just showed you, and quantified the trends over time. And we complemented these imaging data with full lipidome coverage for each lipid probe at each time point. And these types of data then allow you to really write up mass action models of lipid transport. Also, for instance, in this case, we deliver the probe to the plasma membrane, and then you can have vesicular transport along the secretory pathway in the retrograde direction from the plasma membrane to the endosome, to the Golgi, and to the ER. And you can have non-vesicular exchange between the plasma membrane and the ER directly, and between the ER and the mitochondria. There's a couple of a priori assumptions that go into this model. One is that the transport along the secretory pathway is in fact vesicular, and I think that's a reasonable assumption to make. And the second one is that exchange directly between the plasma membrane and the ER has to be non-vesicular, as there is no known vesicular pathway that directly connects these two organelles without first going at least to the Golgi apparatus. And the same thing holds true for the ER and the mitochondria. Now, these individual arrows reflect elementary reactions. So this arrow here basically can be written as transport of material from the plasma membrane to the endosomes. And then essentially you can write up an entire set of differential equations for all of these reactions, and we can fully parameterize these models. And I'm just showing you for the two examples in the first slide. This is how the kinetics look like. For the saturated species, they are relatively slow. So you only reach steady state at around 60 to 80 minutes. Whereas for the polyunsaturated species, this is much, much faster. And having these models parameterized allows you to draw some pretty clear mechanistic conclusions. For instance, you can look at the different transport rates for vesicular transport from the plasma membrane to the endosome, and for non-vesicular transport from the plasma membrane to the ER. And if you quantify that over all lipid species that we checked, you see that on average, non-vesicular transport is about an order of magnitude faster than vesicular transport. And now if we do that a bit more fine-grained and we look at the individual lipid species, what you see is that the major differences between these rates between species are in the non-vesicular route and not in the vesicular route. So we conclude that non-vesicular lipid transport is both faster but also much more specific than membrane trafficking. The second thing is there's two possibilities how organelle lipid compositions can be maintained. One is by exchanging lipids between each other, so via lipid transport. And the second thing is it could also be by localized metabolism. So what we also acquired are kinetics of bulk metabolism. What I'm showing you here is just the fraction of the initial lipid species that's still present in the sample as a fraction of the total bifunctional lipids. That means you have initially, of course, almost everything is the species that you supply, but then over time, the bifunctional fatty acid gets incorporated into other lipid species. So the bifunctional lipidome expands in complexity. And basically how much of the initial molecule is still left is a proxy for the bulk metabolic rate. And this you can then essentially nicely fit with a mono-exponential fit. And if you compare the ratio of the transport, of the rates of transport and the rates of lipid metabolism, what we find is that lipid transport is about 10 to 60 times faster than lipid metabolism. And that in turn means that non-vesicular transport events occur at a much, much higher frequency than metabolic conversions. Now, non-vesicular lipid transport alone doesn't set organelle lipid compositions, because if you have two populations in vesicles and lipid transfer protein, the only thing without an additional energy input would be that the lipid compositions equilibrate. And we know that the lipid compositions of all cellular organelles are very, very distinct. So if we make the finding that the fastest process in the entire network is non-vesicular lipid transport, there needs to be an energy input and there needs to be directionality in it. And one hypothesis that has been put forward in the field is that this is via coupling of transbilayer lipid movement to interorganal lipid transport. And the plasma membrane, but also other membranes of the secretory pathway contain so-called flipases that generate a transbilayer lipid gradient, which is called lipid asymmetry. And the energy stored in this gradient could be used to drive interorganal lipid transport. So these flipases and scramblases could funnel lipids directly into the lipid transfer proteins, and in this manner generate a directionality in lipid transport. And what we did was, of course, we genetically knocked down the common subunit of these P4 ATPases, so you can't make a full knockout, that's lethal, and looked at transport of the substrate lipid phosphatidylethanolamine. And what you see here is the comparison of phosphatidylethanolamine transport in wild-type cells and in these knockdown cells. And it's immediately clear from the raw images is that transport is much slower in the knockdown cells. So that's number one. But if you quantify it, you can also see that the steady state levels start to change. Also, you can calculate a quasi-equilibrium constant, and that is very different in the cells where there's a lot less of this TMM38 protein. And this change in steady state levels, that is the hallmark of a perturbation of an active transport. So we conclude that lipid asymmetry is a key driver of directional lipid transport. And the main discovery that we made is that lipid asymmetry-driven non-vesicular lipid transport is a primary, if not the primary mechanism for the maintenance of organelle lipid compositions. With that, I thank everybody in attendance. It's a pleasure to speak. And of course, I thank my team, great people to work with, collaborators, the facilities of MPI-CBG who make all of this possible, and also the funding agencies. Thank you for your attention.
Thanks so much for the talk. So I think we have some time for question. So first, maybe I can ask a question.
Mm-hmm.
So I think that the lipid labeling is very as a technique. It seems to be also from the literature very noisy, but actually you showed this like this very beautiful picture. So I was thinking, what is like the secret for that?
Okay, 5 years of tech development. So it was not the one advancement that we made. Of course, efficient probe delivery is one. We don't use commercial UV photoreactors, so we build our own LED-based photoreactors that allow us to basically directly irradiate through the bottom of the microscopy dish at a distance of like probably 2 to 3 millimeters. Then much shorter pulses. So this here is something like 3 seconds irradiation and then immediate fixation. Usually, when you have these much longer photo irradiation periods, then you get a lot of tissue damage and so on. And I mean, of course, the main advantage here is that these things are treated like relatively normal lipids by the cell. Also, we did a lot of controls. We did a lot of controls in terms of what is the effect of these diazarine and alkyne groups, and it turns out that's about 1 additional double bond. Also, I mean, there's a very long supplementary information, and we put every detail in there, but it really took a long time to optimize this protocol. So it's not the one advancement, but you have to put it all together, and then you also get decent data quality.
I see. Thanks so much. I think we have another question. So
yeah, tunnel LTPs. I think so. What the respective flow rates are, I don't know yet. I think there's just a structure out where you actually have one of these scramble lasers and a tunnel bridge-like LTP in a complex by Karen Reinisch's lab. So I think in terms of specificity, and I'm guessing, no, is probably going to be encoded in the adapter proteins. So in the flipase scramblase part and maybe additional adapters. I don't think that a bridge-like lipid transfer protein alone can give directionality or a lot of specificity. So that would be my take on it. I am not aware of a data set that shows that in the cell. Broad accessibility. Yeah, so we wrote a grant for a startup that didn't get funded. Currently, the best thing is to write me a mail. You have Milena at EPFL, who's also working with similar probes. We, of course, share the design of the photoreactors. We share probes as much as we can. Yeah, but ultimately, it would have to be commercialized. Yeah. I think another couple of high-profile applications, ideally in collaboration, and then maybe the people that have the money get nicer with us. There's a bunch of questions. Yeah, it's possible to do the same imaging at tissue levels. Okay. So in vivo, I don't see it yet, simply because the amount of probe that you need is just too high. Also, it's a practicality issue. At tissue level, we have done it in organoids.
So basically, people are not seeing the question that you.
Okay. So I have to. So question by Ficado: is it possible to do the same imaging at tissue levels or in vivo? In vivo, I think it's practicality issue that you would have to load tons of probe that simply doesn't exist. In tissue organoids, yes, that works. And tissue sections, I also think that should be doable. So the next question is whether there is a whether we are able to observe lipid composition among diverse ER domains. And whether there's an effective method to check this with bifunctional lipid probes. So we can, of course, look at partitioning of individual lipid species. And that is actually one of the projects in the lab to look at that. But of course, overall lipid composition would mean that you have to have mass spec imaging at subcellular resolution, which is, unless Giovanni tells me that it's in development, not something that I'm seeing right now happening.
Sorry, Andre, to stop you.
Okay, I will answer in the chat.
If you can do it, yeah, that's perfect. Thanks so much.
Okay, good.
So that's great. Thanks for the talk again. Thanks for being here. So
Many thanks.
Here, for the next speaker for today is Theodor Alexandrov. I think he's already here, so I can invite him to take the stage. Theodor.
Yes, I'm here. Yes.
Thanks so much.
Wait, do we start? I mean, you guys get a break. I think now there is 2 minutes break. Do you want to start now?
Well, let's say that the break were kept for buffering time that apparently were taken by questions. So, but yeah.
I mean, ask your breaker.
Thanks so much.
Okay, cool. Yeah, thank you once again. Thank you to everyone who organized this fantastic day, already second time and second year. And thank you guys so much for getting really community together. and for the opportunity to present us some of the work that we do in spatial and single-cell metabolomics. My name is Theodore, I go by Theo, and today I'll present our research team. And we also recently created a startup deep site on single-cell metabolomics. And but let me show first really our team and like really the heroes who moved from all the way from EMBL to University of California, San Diego last year and also this year. And I'll be presenting their work on their behalf today. So what we do, we develop technologies mainly for spatial and single-cell metabolomics. That's why we're really, really excited to be here today. Using imaging spectrometry, focusing on metabolomics, but also other omics. And often we have some twist with big data and software. And this is our lab manager, Shabraz, and you might know him, and also Gianni from the recent paper on high throughput SpaceM, and Lucas, software developer. You probably, if you use Metaspace, that you've heard from Lucas because he's supporting and everything, and he's our main software developer there. Louisa, PhD student, and she's had a really, really exciting project on T cells that I'll not be showing today. Shabrat, who is working on PhD student, working on multi-omics, single-cell multi-omics, and Bishoy, he's a senior PhD student, defending his PhD thesis actually tomorrow, and he'll also present later today. So, and what we are focusing here on metabolomics and metabolism, and by now we all know that metabolites, they're so important and so useful because they're closest readout to phenotype. But also, as Martin presented in his first talk, there was sort of like shift in attention and acknowledgement of the roles of metabolites as being in the totality as metabolome as a regulatory hub. And this was also discovered in lipids, but also for small molecules. So, and this really puts metabolism into the spotlight. And as was really, really nicely put by Craig Thompson, by Catherine Bellin, more than 10 years ago. So central dogma is not really just one-way street, but and not only metabolites have their close readouts to phenotype and function, but they actually, it's really a two-way street where metabolites play a key role as activity metabolites or regulatory and signaling metabolites. So this really puts metabolomics into the spotlight as omics able to profile metabolome. But for me as technology developer, it's really interesting to reflect on other advantages of metabolomics, which are really, really strong and I think important for the community today. And these are what we have on our hands is actually the most scalable, the highest throughput, the most affordable single-cell omics. And if you really reflect on this and compare how, for example, matches versus single-cell RNA-seq, that you will see that it's actually already has higher throughput. Although our metabolomics, single-cell metabolomics is so much younger field, but it's already so much faster. And in terms of cost, it's incomparable. So what you now can do with single-cell metabolomics, it is impossible to do it with single-cell RNA-seq just because of its cost prohibitive. And what's really cool now, there are even technologies and studies showing that it's combinable with protein RNA detection. So we really have this amazing technology, so powerful and so affordable on our hands. And now using the power of this technology, can we now start discovering metabolic states? And Joelia has mentioned that in transcriptomics, now there are discussions about 5,000 of metabolic states only in the brain, right? And Joelia and Giovanni, they actually had this really interesting opinion piece recently where they focused on lipotype hypothesis, that these groups of cells with a similar lipids, not only they can be discriminated and have distinct composition, but they also have some important drivers of functional relevance of cell fate. So now the question is, can we expand it? And can we use single-cell metabolomics to discover metabolic states beyond these lipotypes? And this is one of the key goals and strategic goals for our team. And what we use for this, we again develop technologies and also apply technologies based on using multi-imaging mass spectrometry. Probably everyone here in the room knows this. This is amazing technology which can generate a lot of data in spatial resolved manner. And what's really, really cool is now commercially available technology can do 5 to 10 micrometer pixel size analysis, which gets us approaching to a single cell resolution, in particular for cultured cells. So we've been working in this field for some time, initially focusing on the key bottleneck in spatial metabolomics, which is molecular identification. because we can do visualization, stat analysis, but we need to move to molecular validation interpretation, and we need to solve this bottleneck. And also Martin, for example, really nicely highlighted some of the challenges like in-source fragmentation, and also that we should not just do exact map search in, say, against lipid maps. And then in our paper where we proposed the solution metaspace, we have shown why we should not do this, because if you do exact map search, then your false positive rate will be above 90%. And it's definitely not something that you want to do. So, so we developed over the years, and we are so excited that it really got good use, software called Metaspace, which is algorithm for metabolite ID, but now much more quality control, visualization, analysis, data sharing. It's all in the cloud. It's all in a browser. It's also all open source. And now there is a lot of thousands of users using it, and there is hundreds of publications. And Lucas, software developer, and Bishoy, data scientist, they are really now the key people behind this. And when people started using Metaspace, we were super excited that many people started sharing their data publicly with others. So now we have, out of these submissions, we have 15,000 public data sets. So we realized that we're pretty much sitting on a gold mine. But what can we do with this gold mine? What can we learn? Of course, we want to learn those metabolic states and like really biology, but it's very tricky, right? Because these data sets, they come from different sources. We don't even know details sometimes about the biological system, perturbations, and so on. So first, before moving to real discovery of metabolic states, we thought, can we improve the technology? And this is what we did. This work was led by Bishoy, who will be later presenting another project. And here I'm showing it because it's really nicely showing this, how big public data can be used. What Bishoy and others, they have done, they took public data from Metaspace. They took two subsets from animal and plant data sets. And afterwards, they came up with a machine learning framework to retrain machine learning model that would actually be discriminating good ions from bad ions, they target from decoys. And through this, really learning kind of like adaptive score that can do identification better than rule-based score we had before all these years. And we call it Metaspace ML. And now this model is available to everyone. And we use it, and we were surprised recently to check how many users started using it. So look at what we've done here. So we initiated first submissions to Metaspace. We kind of encouraged people to make data public. Now we use it for training ML model, which can detect more metabolites. We provide it back to users, and we hope that this will lead to even more submissions to Metaspace, which ultimately we can retrain this model when we have even more public data. So once again, I'm showing this to really emphasize how important now in the age of AI ML to share your data publicly, because by doing this, this is really the case study how this will improve the tools that maybe you will be using afterwards. And another announcement that we are very, very happy to make is that to really improve the sustainability of Metaspace, recently we split Metaspace into two versions, academic and pro. And academic is free, it's open source, it really supports public data. And pro version is subscription-based, and it supports private data and has all other advanced features which are necessary for this, for the pro analysis of the data. And by splitting this, we actually improving sustainability of both academic and the professional version. And you can check out our LinkedIn or website. There is actually so many features, new features coming. And Lucas and Bishoy are involved, and Bishoy is actually working full-time now on Metaspace Pro. So long story short, so let's now switch the gears toward the second topic of this beautiful online day. And it's about single cell, right, metabolomics. And some time ago, so I had an opportunity to reflect on the progress of the spatial metabolomics. And what I have realized that not only it became not a niche field anymore, but it really became the driver of innovation, because by addressing so many challenges in spatial metabolomics that do not exist in bulk metabolomics, so the vendors and the whole community had to really improve the technology a lot, improve sensitivity, improve data throughput. Some of the aspects which were generating data for spatial they were generating at such fast pace that vendors had to change completely everything what they're doing on the back end. So, and through this, it really enabled new technologies, in particular spatial single-cell metabolomics, but also others like high-throughput screening, pharma, intraoperative mass spec, spatial fluxomics, and so on and so forth. And for us, this is exactly what happened. So we kind of like jumped into this emerging field of single-cell metabolomics, and with our first PhD student, Luca Rapaz, and since then we have developed, so to say, a family of methods for single-cell metabolomics. Spatial initially used for lipids, actually. So then afterwards, 13C Spatial for single-cell flux analysis, also for lipids. And only recently we really cracked how to detect small molecule metabolites. And now we have this high throughput and small molecule metabolites methods. In principle, they all work in a similar manner, that we take cells, we plate them on a glass slide, then we do microscopy to find where the cells are, but also morphologically, phenotype them, then detect metabolites, lipids, now in protein markers, then overlay these two types of data with cellular precision, and now we have our single cell profiles. And once again, I really want to emphasize how cool is that this technology is about 10 times faster and 100 times cheaper, roughly, than single-cell RNA-seq, and at the same time detects all these relevant profiles. And why do we know that these profiles are relevant? I want to show you just excerpt from this paper. And in this paper, Gianni and Shiraz from our team, they developed this high-throughput method, and then afterwards, they analyzed a bunch of cancer cell lines from so-called NCI60 panel, and also HeLa. And if we now look at the UMAP, where one dot is one cell, we have a bunch of cells here. We have here around 200 metabolite ions. And for every cell, we detect maybe 100. But because there is diversity, also metabolic diversity between cell lines, in totality, we're detecting 200. And now we can do all the analysis, which is normally done in single-cell RNA-seq, for example, like finding markers. And it's cool to see here that we can find markers, for example, for this particular cell line, which were detected or expressed only in a very small percentage of cells. And it would be definitely diluted in bulk analysis because they were detected in less than 20% of cells. And but at this point, you can say that, okay, I mean, it's all nice. But I looked at here, there are distinct differences, and most of these markers, they expressed in the majority of cells. Why do we really need single-cell metabolomics? I mean, we probably biologically could have achieved these results also with bulk. So let me show you one method that we also published in the same paper, which really shows the potential and unique advantage of single-cell metabolomics. So for this, what we did, we considered HeLa cells. And then we treated cells with a 2-deoxyglucose. And 2-deoxyglucose is sort of analog of glucose. It also gets transported inside cytoplasm, then it gets phosphorylated by the same hexokinase, which is phosphorylase glucose. But then this molecule doesn't get metabolized further, so it actually gets accumulated because hexokinase still works. And how general glycolysis works is actually like glucose phosphate, or in this case, this analog, it starts inhibiting hexokinase, so it's actually inhibits the whole glycolysis in the upper part. So it is very common inhibitor of glycolysis. So now we take HeLa cells, control, and we take HeLa cells treated for 12 hours at 24, and we see distinct difference in the metabolic profile. And if we look at the markers, we see that glucose gets accumulated, which is expected because we're blocking this step, but transport is still working. We see some metabolic intermediates, they drop down. We see some also respiratory metabolites like citric acid, they go up, which potentially compensatory mechanism. So it's all kind of like expected, but again, you can get the same results with bulk metabolites. Now let's do truly single-cell analysis. So let's now, for 2 metabolites, calculate the correlation between them. So one dot here is one cell, there's such metabolites. If there is a correlation, means that there is some sort of regulatory mechanism between these 2 metabolites, which really regulates the intensity very precisely across all cells. And now what we can do for every 2 metabolites, we can see their pair, and we can draw a network, right? Where dot and metabolite and line connects them if they are here co-abundant. And it's really cool to see what we have here. So first of all, amino acids, they actually in one block. Nucleotide precursors, they're in another block. But let's now compare the conditions. These are treated cells, and these are untreated cells. And if we start comparing this network, we see that it's actually pretty cool because glucose was not here at all. But upon inhibition, gets accumulated, and it's actually becomes a central metabolite co-regulated with amino acid metabolism, in particular critical metabolites like glutamine and glutamine. And we see that if we do like stronger treatment, this still kind of this mechanism still preserved, and we have even other approaches, other findings. So, and I want to emphasize that this results is almost impossible to get with bulk. So we exploit natively present cell-to-cell heterogeneity in large N to find the correlations. We reveal relationships between metabolites from distant pathways. So it's not only enzymatic relationships. And once again, so it can reveal results which previously would require either flux analysis, which is possible, but tricky to do in experimentally tracing, or extensive perturbations, either genetic or non-genetic, so that you can actually get that N. And what we're doing here, we're really working with cells in their physiological heterogeneity and just learning from the biology which exists, in this case, of course, in vitro. Okay, but we spoke a little bit about heterogeneity, and I think I have one more minute to present one more result here. So what about heterogeneity here? We actually don't see it much. We see mainly the conditions which are different between each other. So do we have actually heterogeneity within the condition? So, and if we start clustering, this is a condition, but this is clustering. We notice that this group of cells, they actually clustered not by their condition, but by something else. And we can find markers of this. So, and if we look at this, it's really spatially heterogeneous. So there is sort of like two different types of cells here, which are spatially heterogeneous, and they are not just the timing of treatment. So what we did next here is we clustered every conditions separately. And what was cool to really see that this clustering actually goes through the middle of all these clusters. Here, this one, this one, this one, this one, this one, this one. And if we find markers of this cluster versus this versus this, versus this versus this, then we see pretty much exactly the same markers. So we see, for example, fatty acids here, fatty acids here, fatty acids here. We see citric acid here, or nucleotides here, nucleotides here. We do not have validation of this, but we think that this may be heterogeneity caused by the cell cycle. So, and what's cool is that now we can see this heterogeneity, which is preserved through metabolic perturbation stimuli, and 2D is a pretty strong one, and can be associated with cell types, cell cycle, and other phenomena. And just to wrap up, I would like to show where we think it can go in the future. And this is a screenshot from our review from Nicola Zamboni that is accepted, soon will be out, where we are brainstorming about convergence of single cell metabolomics into high throughput and population scale metabolomics, not only to reveal these metabolic states, which are likely conserved, at least some of them, across different cell types, but also kind of converge by merging heterogeneity on the cells and diversity on the populations to really create data for AI, ML, and really address the problems that can help build comprehensive atlas of human metabolism with cellular precision, population-wide scope, metabolic heterogeneity, and population diversity to reveal disease signatures where this population scale metabolomics is so powerful. And of course, interrogate non-genetic factors and enable data to create metabolism component of virtual cells. And with this, I'd like to wrap up. Thank you so much for your attention. The whole team who contributed from Amble, but now at UCSD, our MetaCloud and Metaspace Pro team, DeepSide. And I also want to emphasize that Mel Bailey, who is presenting later, she's organizing a really cool conference on single cell mass spec that will be next year in London. Thank you so much for your attention.
Thank you for amazing talk. I think we have questions in the chat, and you can answer there because we are short on time. So our next speaker is Carsten Kopp and Lars Gruber from Mannheim University of Applied Sciences.
Thank you so much.
So I thank you.
Yes, I will quickly share my screen. So hopefully you should all see the presentation now.
Yes. Perfect.
And hopefully still the presentation.
Yeah, and Lars and I will do it together. I'll just introduce us very briefly on the next slide. If you already switch Lars, and then Lars will take you through some contents actually. So, we're sitting in Mannheim, Germany, running the CMOS Center here, which is involved in mass spec imaging quite a bit, coming more and more to single cell. We run an industrial consortium and are a partner side of public drug discovery, EU Open Screen. Yeah, and working with a lot of different instruments. We have tackled on the next slide cell assays for a number of years, starting about 10 years ago, where we designed multi-mass spec-based assays on 384 well plates. And that has recently morphed into 3D cell culture approaches, but also into single cell approaches that we want to talk about today. And that is mainly Lars's work, and therefore he will present that himself.
Okay, yes. Today I will talk about the single cell analysis, which we carried out in the scope of our recent paper here. So, thanks to my thanks to the talk, thanks to the talks before, I do not have to introduce multi-MSI. So yes, I guess everybody in the audience is familiar with it. Good, but I also think that not everybody in the audience is familiar with quantum cascade laser mid-infrared imaging microscopy. So let's start with a quick comparison of QCL-MIR imaging and FTIR imaging, which is, I guess, the common way of infrared imaging. So the main difference between QCL and FTIR imaging is the light source. In QCL-MIR imaging, we use a tunable laser source, whereas FTIR imaging relies on a broadband light source and the so-called Michelson interferometer. So, but the results are the same. You always get your absorbance or transmittance spectra here, and you can assign specific bonds there to molecule classes like carbohydrates, nucleic acids, proteins, or lipids. But then the question would come up, why do we have to use QCL-MIR? So here I can show you in more detail why QCL-MIR is superior to FTIR imaging. So in QCL-MIR imaging, we use a wave number sweep across the entire field of view here and measure directly the hyperspectral cube. Whereas in FTIR, you have the pixel by pixel measurements of the interferograms, which are later Fourier transformed to your wave number spectra. So as you can see, this takes a lot of more time. And for example, to quantify this, we estimated that we can measure over 8,000 pixels per second with the QCL-MIR imaging system, whereas we measure 50 pixels with FTIR imaging. So the only disadvantage of the QCL-MIR imaging is the limited wave number range. So in QCL-MIR imaging, you are not able to measure above 1,800 reciprocal centimeters. So you only have the fingerprint region. But you can still take advantage of this measurement, especially here in the case of motor neurons in mouse spinal cord. Here you can see an MIR image of a mouse spinal cord and measured at the amide-2 bond. We then generated a mask of the gray matter in the spinal cord, as you can see here, and tried to extract the motor neurons there. So as you can see here, we did it based on specific wave numbers. For example, here we used the sum from 1710 to 1730. And for contrast reasons, we relied on the first derivative of transmittance. And here you can see the difference of the gray matter area compared to the motor neurons. And the result is the following. We were able to specifically target only the motor neurons in the gray matter area of the spinal cord. So basically, this is the first proof of our concept of QCL MIR imaging guided multi-mass spectrometry imaging of single cells. We then aimed for a more, let's say, biologically relevant study. And therefore, first of all, here you can see that we also did this based on n equals 4 biological replicates. We use here the phosphatidylinositol lipid as a marker, and you can also see that we did this in comparison with an H&E staining to really verify that we addressed the motor neurons and not something else. Good. But now let's go on to our biological case. We used the experimental autoimmune encephalomyelitis murine model for multiple sclerosis. In this murine model, the lesions in the white matter area of the spinal cord are induced to simulate the multiple sclerosis effects. And in QCL MIR imaging microscopy images, you can see here that we can also identify these lesions here in the white matter. So for example, here, here, and here. And still here you can see how we identified the motor neurons. So when we now compare this with our multi-MSI data, we can also see that these lesions also show intensity for given features in that regions. And then we compare this also with an H&E staining. And you can see here that the cell density in this lesion area is quite higher. But then we thought, why do we not use our QCL MIR imaging workflow to only extract these lesion regions and make them accessible for multi-MSI? Because of course, these lesions are really narrow. As you can see here, it's only 240 micrometers. So you have to measure multi-MSI at 5 micron pixel size here. And as you all know, 5 micron pixel size means the measurement time is long and the speed is low. So we then applied our QCL MIR imaging workflow. And then based on that, we were able to obtain these lesion regions based on k-means clustering, as you can see here. We used here the EAE spinal cords, generated also the mask for the white matter here, and then extracted only the lesion areas based on our given wave number subset. And just to compare, we also did a similar workflow for the control. We also extracted the white matter area, and then we said, okay, we want to keep this as comparable as possible, and then said we use the same amount of pixels that we extracted for the lesion area and shrink the white matter area in the control to the same region size. This also is just to keep the measurement times reasonable. And the result is what you can see here. Here you see that we were able to successfully extract these regions of interest and validate them by multi-MSI that we hit the lesions. So as you can see here, the lesion is here really in the center of the area of the region of interest. And what we also did, we also artificially increased the measurement area just to have the contrast of lesion and surrounding healthy area. Just because then we can also do the comparison between here lesion and healthy surrounding area. And here you can see what I told you before. We shrink the white matter control regions of interest to the same size as the lesion area here. And here we relied on a second derivative of a spectral band, which can be assigned to the CH2 scissoring vibration in the lipids. And as I told you before, yes, this here, our QCL MIR guiding guidance brings the advantage that we now make this 5 micrometer study of the lesion regions of interest reasonable in measurement times, even if we have a large scale study with n equals 3 biological replicates, because and the TIMS on mode activated. Because when you, let's say you measure the entire section here in TIMS on at 5 micrometer and TIMS on, I estimated it at about 20 hours of measurement time if the spinal cord is quite large. And imagine if you have these replicate studies, you would block an instrument for an entire week, for example. And now you can, of course, based on the amount of lesions, you can then reduce the measurement time to 1 hour or half an hour. And now let's see, here we have what I told you before, we have the large scale study for the biological replicates. And here we have characteristic molecules which accumulate in the lesion area. So for example, we have here this ceramide phosphate, we have the PE and the PI lipid here. And then we also did the statistical analysis here and saw that especially here, this sphingolipid marker, the ceramide 1 phosphate, is highly upregulated in the lesion areas compared to the control. This is also in line with what was reported in other studies of human multiple sclerosis lesions. And also we see that these membrane lipids are also upregulated. Okay, what we also see is that we do not only find upregulated lipids in these lesion areas, we also see for given lesions that you can detect, let's say, early onset of demyelination caused by the sulfatides. Here we have the most prominent sulfatide. I guess everybody who is a little bit familiar with brain lipids knows this because it's also the most abundant brain sulfatide. And here we can see that in the lesion area, you get slightly decreased intensity already for the sulfatide. And even when you do the statistical analysis, you can see it. Of course, it is still not significant, but it is visible, and we can also, and you can also, and we can, yeah, see it. Okay, what we also did is we also validated all of our biomarkers by imaging PRM passive experiments, meaning that we obtain MS2 spectra for each of them and then annotated the fragments to be sure that these that we annotated the right lipids. Of course, what we also did is we also thought we can do the lesions, but what other targets could we address with our workflow? And then we aimed to extract only the glomeruli from kidney sections, meaning that this also results in a reduced data acquisition by 95%, as you can see here on the right. And here we relied on a ganglioside marker to verify our glomeruli here. And as you can see here, it is also a significant reduction in time and measurement. And also, as I told you before, we also did here the validation by IPRM Parsef, and also we can now here really do nice analysis of CCS value evolution depending on the chain length of the gangliosides. And also, what we also did is we compared the center of gravity from our regions of interest derived from MALDI-MSI and QCL-MSI, and saw that we were able to that we have a Euclidean distance difference of about 20 micron, but since our MALDI-MSI experiments are taught manually, this is quite a good result, because 20 micron shift in manual co-registration is really good. Okay, so with that, I'll come to the conclusion of this talk. And as I presented here, our QCL-based MIR imaging guided MSI workflow offers these high throughput capabilities that I showed you with the example of the spinal cord lesions, but also could be extended to other large-scale studies of small regions of interest and single cells, of course. And also based on our IPRM Parsef workflow, we can identify our biomarkers. And overall said, this workflow really helps to significantly reduce acquisition time and data size. And with this, I come to the end of my talk. I also want to thank the organizers for the opportunity to speak today, and also thank my colleagues here who contributed also to this work.
Thank you so much, Gruber, for the talk. That was very nice. We also have time for a question at least. There is a question in the Q&A, actually, if you want to have a look, or I can maybe actually ask if you want. So, how do you deal with ceramide 1-phosphate annotations, provided the possibility that it comes as a fragmentation product of the sphingomyelin? Is there a check that can be made to control for that?
Yes, of course. You also in-source fragmentation can also be a problem, especially in MALDI-MSI. But at that point, we also, since we do the analysis here, negative ion mode, the sphingomyelin is usually detecting positive ion mode. It makes that not really. It makes it more realistic that it is this C1p lipid, and also in line with the given publications, we can be highly sure that we annotated it correctly.
But if I may add to that, I think that's a very good point, where our entire field needs to pay more attention. And in some cases, the distinction in MALDI-MSI would have to come from the precise fragment spectra, whether you see certain fragmentation products or not. And sometimes the intensity is not high enough to see it in IPRM Parsef. So, we would still recommend, and often do it these days ourselves, do parallel LC-MS as well, and then take it from there. Yeah. But of course, Lars is correct about the ion modes.
Okay, thanks so much. There is maybe another short answer if you can. So you claim that the difference in sulfatides is not significant, while pixel variation can be broad. So it looks like it could actually be significant depending to which statistics you're using.
Yes, I understand the question. Yes, of course. The question is, or the topic, the point is correct. Yes, it depends on the statistic you use. Since we did this statistic across all lesions to avoid any, let's say, outliers or anything, this seems to be non-significant. But of course, if you only, let's say, use the lesions, which where you can see the demyelination already, then it would become significant.
All right. Thanks so much for your talk, really. So now we move to the next speaker, which is Peter Nims. So I think I see him here.
Yes. How can I unshare my screen? Here.
Yeah,
Okay.
yeah, thanks so much.
Hi, Alessandro. I'm trying to share the slides.
I don't know
Okay, now I got the permission. Thank you.
so much.
Okay, so hopefully this shows. It shows well.
That's perfect. Thanks.
All right, great. Well, thank you again for having me. It's wonderful to be back this year again. And congratulations again to the organizers. A big thank you to everyone for bringing us together around metabolomics and single cells. So I will tell you a little bit about a little bit of historical overview of how we got into single cell metabolomics and how we are using the chordate embryo to drive technologies and biology forward by probing the metabolic composition of identified single cells in developing embryos. So my laboratory uses the frog model, Xenopus laevis, as a developmental model, as well as a model for stem cell differentiation. And when we got into this about 13 years ago, I was really amazed by the process of development, how a single cell develops into 2 cells, then 4 cells, 8 cells, 16 cells. And immediately we started thinking about in my lab how various proteomic, metabolomic components in these cells might give rise to various biological outcomes. So the challenge, however, with this model is that, as typically with developing embryos, is that development is very fast. The cells only exist about 15 to 30 minutes. The cells also occupy a complex three-dimensional structure. It's a ball of cells, essentially, you're working with. So these are not flattened tissues. The cells can also migrate long distances, several millimeters, tens of millimeters within the embryo over development. And what's exciting, I think, is how over development, these cells occupy different developmental axes to establish the three primary axes, how these cells occupy and how they give rise to the various tissues at the correct time and the correct place in the embryo. So we have 3 primary axes: left, right, a dorsal, ventral, and animal, and top, anterior, posterior, which we call animal, vegetal. And so what I'll tell you about is how we are now going through the various stages of development to survey metabolic and also proteomic changes as these cells occupy these specific spatiotemporal regions in the developing embryo. So these are just a couple of images to show you how the 1 cell becomes 2, 4, 8, 16 cell embryo is here. I will be mostly focusing on this today. What's really great about this model, the South African clown frog, Xenopus laevis, is that the cells can be identified. So we can actually tell what the identity, the phenotype of the cell is. Moreover, the cells have reproducible tissue fate. So I can tell you that the D11 cell located here is a reproducible precursor to neural tissues, retina, brain, spinal column, whereas this cell down here is a precursor to the epidermis. So one of the key challenges, of course, is obtaining your cells, right? And the contents, and it's not different for us either. So in the very beginning, about 13 years ago, we developed a dissection approach where we were able to manually tease out these identified cells from the live embryo and then go about to do metabolomics. And after that, we've developed microprobe strategies where we precision fabricated capillaries that we can drive into identified cells, very quickly aspirate the content of a single cell, and then go about to our business with mass spectrometry. Now, the good news with microprobe sampling is that you can come back to the same cell multiple times because it doesn't sacrifice the actual cell or the embryo, and that has opened up in vivo single cell analytical capabilities through my lab. I will not talk about that today since we already addressed that last year. Now, with this microprobe sampling, we actually measure much less oxidative stress in the cells. So we believe that this is an excellent way to sample the cell and then read out the actual metabolic contents closer to the native phenotype. Now, over the years, after sampling, we've developed metabolomics and proteomics approaches to characterize cell contents. And so I will today focus on metabolomics exclusively, how we separate identified species and then perform pathway analysis. Now, at the very heart of these measurements is an ultrasensitive capillary electrophoresis mass spectrometry platform that I actually developed when I was a postdoctoral fellow with Jonathan Sweedler. So that was a while ago. And the idea here is that we are able to deposit extremely small amounts of samples into a vial where we can then move a capillary electrophoresis capillary over, inject a small amount of the sample, about 1 to 10 nanoliters, and then we can apply up to 30,000 volts across this capillary to electrophoretically separate out the molecules, in this case metabolites, as we'll talk about. Then we build custom-build ionization interfaces to efficiently ionize them, and we use different kinds of mass spectrometers to detect and identify these species. We've spent about a decade learning how to use this platform, basically for metabolomics and proteomics, ranging from microsampling to separation, to ionization, detection, analysis even, and even data interpretation that can be very tough. Now, unlike imaging technologies, which are amazingly powerful, and I'm very impressed by how the field has developed. Unlike imaging technologies, we actually have the ability to also do separation in the liquid phase. Now, this is not this is not in gas phase separation, this is in the capillary, which means that we have the migration time, we have accurate mass, we have tandem mass spectra that we can leverage to identify metabolites very confidently. And nowadays, we can also add collision cross sections. Chemical separation certainly helps to remove ionization interferences, improves quantification. It adds a lot of unique aspects, and capillary electrophoresis-based separation complements liquid chromatography-based separations. We typically identify about 150 metabolites out of about 400 molecular features in these runs, but obviously because of liquid phase separation, the analyses are much slower. So I will not be talking about thousands of cells measured in this talk. Now, the platform is very sensitive and quantitative, as you can see here, built up for various model metabolite compounds. We can go down to about 25 attomoles with about 10% standard deviation in quantification. So that is very important because that means we can also tell not just what is present in the cell, but how much of those components are present to then design biological experiments. So very early on, back in 2015, we started using this technology to ask the question how these identified cells that give rise to different types of tissue fates may harbor different metabolite content. And this is not a question that was raised at the time. So we basically measure the D11, V11, and this tiny V21 cell hiding at the bottom of the embryo for metabolite content. We relatively quantify the metabolites. And in this volcano plot, you can see that there is quite a significant difference between metabolic composition among these three cell types. We could arrange these metabolites into metabolic networks based on KEGG. And what was really exciting for us was that quantitatively, this was the first example of metabolic cell-to-cell heterogeneity in the live embryo, essentially, along the dorsal, ventral, animal, vegetal, and left-right axis that I will also tell you about. Now, some metabolites are very peculiar in terms of their directional enrichment. So methionine acetylcholine we found enriched in the V11 cell, which is epidermally faded, whereas threonine histidine is enriched in the D11, which is the precursor to the neural tissues. So we quantify the endogenous amounts of the metabolites using calibration curves, as I've showed you earlier. So now we know that these cells harbor yay amounts of metabolites, about 10 picomoles of histidine threonine, 1 picomole of acetylcholine, 20, 30 picomoles of methionine alanine. And once we knew this, then we could start designing experiments, functional experiments, to test whether the differential enrichment has anything to do with the cell fate in the embryo. So we basically prepared authentic chemical standards, and we were able to inject that into the embryo. For example, these that were normally enriched up here, we injected into this cell, and then vice versa. We also label these cell fates fluorescently, which we can do in the embryo. And if you focus here, this is the normal fate of the D11 cell. Neural tissues essentially are the ones that it gives rise to. But the moment we injected these V11 epidermally enriched metabolites, they alter the cell fate to epidermal. We also validated this in a reverse fashion. So we basically discovered metabolites using this experiment series that were able to alter normal stem cell fate decisions in the embryo. So that's about dorsal ventral patterning as one axis. Now, we've also explored left and right heterogeneity along at the single cell level. So for example, at the 8-cell stage, the embryos can be bisected into a left side and a right side. We developed a multi-pronged approach where we could extract metabolites using different chemistries, organic content and pH, organic solvent content and pH levels. And essentially, when we performed this relative quantification among the cells, the left and right cells, we found a number of metabolites that were differentially enriched. For example, there's these 5 metabolites enriched in the left cell. There's another one on the right cell. We designed then functional experiments where we could take these metabolite cocktails, inject them into the correct identified cell, and then we went about to trace various markers that are historically used in developmental biology. And we found that in the control embryo, for example, the cell lineage is about the same length on the left and right side. When we injected the right enriched metabolites into the left cell, we got about the same result. But when we went to the left enriched metabolites that we injected into the right cell, we completely perturbed these clones that arise over development. Now, this is very intriguing because it basically shows that metabolites do more than just bystanders. They're more than just bystanders in a cell. They actively contribute to cell fates, whether that's dorsal, ventral, or left and right. Now, since then, we've gone cell by cell over the embryo, and we've uncovered metabolic gradients, literally, from among these 4 cells, for example. And these are cluster analyses showing these trends. And we started asking the question, where do these metabolite gradients arise from? This is quite an unheard question in developmental biology, where mostly transcripts and proteins have been at the focus. So we designed isotopically labeled experiments where we use isotopically labeled metabolites, injected them, and we basically studied how they are transported across these neighboring cells. And we found that these metabolites are transported between the neighboring cells, but in a unidirectional manner. For example, threonine is very easily passing through between, threonine does not pass through between, for example, the dorsal ventral membrane, but methionine did. Based on isotopic labeling. And then that allowed us to propose that maybe gap junctional connections participate in setting up these gradients. So we also designed pharmacological interventions where we could inhibit or promote these gap junctional proteins. And lo and behold, once we inhibited the gap junctions, the metabolite transport was inhibited. Once we promoted the gap junctional protein activity, we promoted the metabolite transport as confirmed by isotopic labeled metabolites. So these membranes, we think, do more than just separate cells from each other in the embryo. And by the way, the directional transport is really interesting because we can now label these, for example, neighboring cells in different color in terms of what kinds of tissues they give rise once you allow the embryo to develop into a larva. So that cell, the D11, is a precursor to the retina, the brain, spinal cord. The cell next to it has a totally different cell fate, head epidermis. And once we started perturbing these cells, for example, with methionine injection, what we found was that depending on which direction the metabolites are transported, you could now alter one or both of these cells' developmental fate. And that's very exciting because, again, that highlights that metabolites contribute actively to establishing these cell fates. Now, I've alluded to the role of gap junctions and membranes. So more recently, we do a lot of proteomics and single cell analysis also. And so we went to the embryo and we literally manually dissected out these intercellular membranes that separate out these two cells, these two cells, and these two cells. And these are kind of the workflows. We can do this because the embryo is large, the cells are relatively large. It is a learning experience, but obviously we can do this. So now we measured about 800,000 proteins in terms of relative abundance between these dorsal membranes and these ventral membranes. Many of them are actual annotated membrane components. They participate in broad biological processes, and I'm just going to focus on metabolism in the interest of time today. Serine, glycine, threonine metabolism, for example, lights up. And it's very exciting because that's precisely what we found about a decade ago to be able to alter cell fates. So now in light of the metabolites and metabolomic and the proteomic information we have about the cells, as well as the membranes separating the cells. Now we're developing actual hypotheses on how these biochemical mechanisms work. We can track both the protein and the metabolites, so we can now correlate them. If one goes up, does the corresponding metabolite also go up in abundance? And we basically these results, which we're about to submit for peer review, they demonstrate the very first example, to the best of our knowledge, of membrane cell heterogeneity in the embryo, which, as far as we can tell based on preliminary data, do contribute to sustaining the cell-by-cell gradients at the metabolite level that I've spoken about. Since my time is almost up, I'd like to just conclude that single-cell capillary electrophoresis mass spectrometry is a pretty impressive technology. We've been using this for about 13 years now with great success. It enables metabolomics, proteomics, peptidomics. It is qualitative, quantitative. It is fast enough to do high-throughput analyses. It provides deep coverage at the omic level. It certainly complements liquid chromatography at mass spectrometry imaging. There are some analytical advantages to actually doing electrophoresis over chromatography, and I'll be happy to answer questions on that. We have used this tool to answer new kinds of questions regarding cell heterogeneity, stem cell differentiation, embryonic patterning, also neuron-to-neuron differences in the mouse brain, but I don't have time for that today. To talk about that today. And so it's certainly a technology that's worth keeping in mind. I want to thank my group and all the funding agencies that make all of this work possible. And again, thank you for your attention. I'll be happy to take any questions. And again, thank you for joining today, and congrats again to the organizers.
Thanks so much, Peter, for the nice talk. We have a question for the audience. So metabolites and metabolite gradients influencing development is such a powerful concept. How do you envisage metabolites instruct cell fate at a mechanistic level? This is a very interesting one.
Yeah, that's a great question. So we have done follow-up studies that I didn't really have time to talk about today using isotopic labeling, proteomics, and essentially we now think that these metabolites perturb post-translational modifications on proteins. We have found evidence for epigenetic modifications that they could contribute to. So that might be the mechanism of action, but it's absolutely an untapped field at this point. There's not much known about their roles. So we are literally laying down this foundation brick by brick, this entire field. So that's kind of exciting. Yeah. So to summarize, we believe it's post-translation modifications, epigenetic modifications on proteins that are hopefully eventually we can link to classical morphogens discovered during embryonic patterning.
Okay, thank you so much, really. If there are other questions, you guys can post it also in the Q&A. So now,
thank you.
Thank you again. So I see the next speaker is Melanie Bailey. I see the Melanie on online already. Melanie, can you hear us? Melanie, you're muted.
Hello, I can hear you. Can you see my presentation?
Yeah, put it in presentation mode.
Are we there?
Well, we can see actually the.
Do I need to swap?
Yeah. Maybe you can actually share the screen and then put it like full. Yeah.
That way. Does that work for you?
No. Okay. Yeah. Here we go. Perfect.
Yep. Perfect. Okay. Well, thank you so much for the organisers for giving me an opportunity to come and talk at this meeting. Apologies if I'm a little bit overdressed for the meeting. We're actually in the middle of our Christmas party here in London. What I actually want to talk to you about today is the seismic facility for single and subcellular omics. So we have recently moved from the University of Surrey to King's College London. So I guess a little bit different to some differences and some similarities with the technology that Peter has just introduced to you with capillary sampling and capillary electrophoresis. We're, I guess, coming at this from a slightly different angle. So I have established a BBSRC national research facility for spatially resolved single and subcellular analysis in the UK. And this is a collaboration between various different people that are doing different omics analysis, bioscientists with applications, and then people that do single cell sampling. And we're supporting around 30 different research groups around the UK. We're co-developing instrumentation from Yokogawa Corporation. And so this is a spinning disc confocal microscope with live cell imaging. So your cells are under controlled temperature and humidity at the point of sampling. It's suitable for analysing cells in a 2D culture, but you've also got fluorescence attachments so that you can get really beautiful images of the cell morphology. Then there's a robot that actually will come down and pick a single cell into a capillary. So it's a very sort of automated approach for capillary sampling. So here's the sorts of things that we can do with it. So first of all, we can pick individual cells into capillaries. So on the left, you can see the shadow of the capillary tip over a cell and a before and after picture. And even if the cell is relatively, or if the cell culture is relatively confluent, we've tried this on lots of different cell lines now with lots of different users, it's possible to pick it, leaving behind the position of the neighbours. And so that enables you to get that spatial information that you can't do with microfluidics type techniques. So not only can we go at the single cell level, but we can actually pick at the subcellular level. So here you can see sampling of the nucleolus. So we take our capillary, we pierce through the cell membrane, we pierce again through the nuclear membrane, and then we aspirate the nucleolus from the cell. We can also pick from the mitochondrial region of the cell. So here you can see the mitochondria have been stained with, I think this is mitobrilliant, and you can see before and after picking mitochondrial samples. And of course, we can use the confocal microscope to look inside the tip to check that we've collected a sample and look for the fluorescent mitochondria. On this slide, you can see sampling of intracellular bacteria. So you can see here a before and after picture of sampling bacteria from within a cell. We can use the sampling system to take them out. We can also sample stress granules that form upon virus formation, which is of interest to a lot of our users. So basically what we've got here is a platform technique that allows you to take bits of cells or whole cells and put them into capillaries. And then that opens up a lot of opportunities for analysis. So what we've been working on for some time in my group now is looking at how can we transfer that material from the capillary onto a surface, into a plate or into a vial, so that we can perform various different types of mass spectrometry. And so that opens up the possibility to look at metabolites, elements, lipids, proteins, and we've got some work ongoing looking at also at RNA. So we've published quite a few papers on using these techniques to look for lipids. And if you're interested, you can obviously go and look at those in the literature. We spent a lot of time looking at, I guess, differences between cells and blanks to try to optimize our methodology to make sure that we're only considering lipids within a cell. We've been able to show that single cell lipidomics conducted in this way. So what we do is we transfer the sample from a capillary tip into a vial and perform LC-MS analysis. We can actually detect around 400 different lipids within our single cells, depending on the type of mass spectrometry technique that we use. And the lipidomics method is sensitive enough to detect a biological response. So we've done things like looking at, with lots of different users, looking at I don't know, different types of cell stress, looking at different drug uptake and the effect that they have on lipid profiles, or looking at different cell types and their different lipid composition. We've also been developing techniques that will allow us to capillary sample cells and then look at proteins or look at elements or metals through ICP-MS analysis. But since this is a single cell metabolomics meeting, I thought I'd focus on the analysis of metabolites using this kind of sampling technique. So I guess the driver for us for establishing this method was one of my collaborators, Danny Best, who is an expert in microbial metabolism, and in particular, Mycobacterium tuberculosis. Now, you might be aware that one of the leading causes of death worldwide is tuberculosis. It's actually the leading cause of death from an infectious disease. And one of the key determinants of infection outcome is the metabolic crosstalk between the bacteria and the host cell. And so Danny is particularly interested in single cell metabolomics to try to understand, why is it that some cells get infected and others don't? Do they have a particular metabolic phenotype? Is that why they don't become infected? And then what happens after a cell is infected? So can we try to dissect some of this host pathogen behavior? Now, if you take a population of cells, the bacteria that you can see here, so this is a fluorescently labeled, this isn't actually tuberculosis, this is BCG, which is the vaccine that we're all given against tuberculosis. And you can see it going in and infecting the cell, it's fluorescently labeled. So tuberculosis or BCG doesn't actually infect, or any infections don't actually infect every single cell in the population, and infection rates can actually be quite low, something like 40%. So if we probe the metabolism of cells in bulk, we're not really understanding the differences between infected cells and their neighbours. We don't really get the metabolic phenotype of infected cells. And so this prompted us to develop a method for single cell metabolomics. So I guess the first challenge is trying to figure out how can you do the metabolomics analysis. And one option for analysing capillary samples is that you can spray the material directly into the mass spectrometer. But this carries some disadvantages because all of your analytes are being ionized at the same time. So they're all competing with each other for ionization. And what we also found is that although we add isotopically labeled internal standards to the back of the tip, so behind the cell, we'll add a solvent that contains our internal standards, it's quite difficult to get those to mix effectively within the cell. So even if we centrifuge the tip after we have picked the cell, then we don't think they're mixing very well. We can see that the cell material is front loaded, and so we're seeing that first, and then we see the internal standard come out. And so the result is that when you do nanosphere ionization, it's quite difficult to see the differences. So you can see here in the principal component analysis to see the differences between blanks, controls, and infected cells. However, what we've actually done is spent rather a long time developing a method to sample cells into tips, add our internal standard, and then transfer them into vials, and then perform LCMS analysis using a Hillick chromatography method that we've developed. And here you can see that now we can get really nice differences between our blanks, our control cells, and our infected cells. So we're specifically sampling cells where we can see the bacteria and we can see that they're infected. And so here you can just see in the heat map, we've got really nice clear differences between infected cells and blanks, or cells and blanks, but also between control and infected cells. And so we can start to pick apart some of these differences. We did actually target this method towards amino acids, and we can actually see upregulation of certain amino acids in infected cells. And this is actually consistent with previous literature that documents redox adaptation of macrophages in response to infection. So moving on from this, we collaborated with Thermo in San Jose, and we were given an amazing opportunity to use one of their newest mass spectrometers before it came on the market. So this is the Orbitrap Exedium Pro, which is now actually available, but at the time it wasn't. And we scaled down the methods to nanoflow. So one of the things that I haven't mentioned is the data that I just presented is at analytical flow rates. So our sensitivity isn't sufficient for us to be able to generate MSMS spectra, right? So we're only able to tentatively identify analytes through their retention time and their accurate mass. However, using the nanoflow methods that we created at Thermo, and probably to an extent the new mass spectrometer, because of the enhanced ion transmission and all the rest of it, faster Orbitrap, we're actually able to generate significant numbers of annotations of metabolites based on, we've sort of categorized them based on their M/Z value, whether we can assign them a formula, and then how many we can actually name, named compounds using the HMDB. And we used two different methods. So one was a Hillick method, but we also had the opportunity to collaborate with others who had developed an ion pair reagent methods. And here you can see these two methods actually give you really nice complementary overlap of metabolites. And so using them together, you get quite a nice coverage of the metabolome. So this is just showing you some data from the two different methods. So what we did here was we picked control cells that had never seen an infection, infected cells, so cells where we could see the bacteria in the microscope image, and then bystander cells, so cells that had no bacteria in them but were sitting near an infected cell. And so you can see that firstly we're able to see the differences between control and infected cells, but also look at these interesting responses in the bystanders. And obviously using the two different methods, we see slightly different things, but we're seeing differences in sugars, amino acids, TCA cycle metabolites, all sorts of things. And so this is a really interesting problem that we're now trying to unpick. In terms of the technology, I think what's really exciting about this is that we were actually able to take a cell that we had picked and then analyze half of it with one method and half of it with the other method. So we're actually able, we have enough sensitivity to see half a cell and detect some metabolites, but also see differences between bystanders, control, and infected cells. So in conclusion, we're developing single and subcellular capillary sampling and coupling that with LCMS. We can perform live cell imaging followed by single cell metabolomics using the Okagawa instrument. And we found that the Exedium Pro gives excellent coverage of single cell metabolites. And we've actually installed a Exedium Pro, but also an Orbitrap Astral in our lab recently, which we're developing methods on for both lipidomics and metabolomics at single cell level. So looking forward to reporting on that soon. I also wanted to let you know, I'm sorry if somebody has done this already. I haven't been at the start of the meeting, but I just wanted to make everybody aware of an exciting event. So the International Single Cell Mass Spectrometry Conference, which was in Chicago last year, Denmark the year before, and America the 2 years before that, is coming to London. So please put the 6th to the 9th of October in your diaries for a brilliant conference. The last few conferences have been absolutely awesome. It's a mix-up of proteomics, metabolomics, and we're actually including elemental single cell mass spec in this conference as well. Yeah, please do come and join us. And finally, I'd like to thank all the wonderful people in my group that did the work that I presented, the funders, and all our collaborators. And thank you so much for listening. Thank you.
Okay, Melanie, thanks so much for the nice talk. So there is, I think, one question from the audience. So the question is, fantastic. TB infection is associated with tag accumulation. How does the metabolic rewiring you observe relates or cause that?
Yeah, good question. So we do not measure tags in our methods because we're just looking at hydrophilic metabolites, but we do have a lipid method that detects tags, and we have detected tags before in single cell, so it might be interesting to have a look at that in future work. Yeah, thank you for your question.
Okay, thanks so much. So we're a bit in advance, but that's perfect. I think we can just go to the next speaker that is already here, I see. Which is Paola Tasca from Yara Lab. Paola, can you hear us?
Yes, can you hear me?
Yes, that's perfect. You can share the screen. Okay, that's perfect. Thanks.
Yes. Hello, thank you for the opportunity to share with you my work. This is still unpublished, so hopefully you will see it soon. I'm Paola, a PhD candidate in the metabolomic groups of Martin-Gera, the UMC in the Netherlands. And we have been working together with Yassin Mohammed on designing a computational pipeline that addresses some of the main challenges in mass spectrometry imaging data analysis. I see everyone here is an expert on MSI, so bear with me. This is for my friends on YouTube. What we do in MALDI MSI, after sectioning the tissue, we apply chemical metrics that facilitate the ionization of the molecules at the surface. In our case, we do metabolites and lipids. We then define a grid of spatial coordinates that instructs the laser of the mass spectrometer where to shoot. And when the laser hits each coordinate, each spot, the molecules at that position are ionized indeed, and their masses are recorded in a mass spectrum. So, for example, if we measure a whole sagittal section of mouse brain at 10 micrometer spatial resolution, we will get more than 600,000 spectra. And if we go up in spatial resolution, so even at the now we can do 1 micrometer in subcellular level, we really obtain a huge increase in the number of spectra. So how do we visualize this data? We can either select each position and look at the molecular content of that position, or we can select single molecules from the average spectrum and visualize the spatial distribution of that. Moving to the data analysis, every group has its own pipeline, but if we consider the commercially available software skills, the analysis typically begins with peak picking, which can be done manually on the average spectrum, or now we can use the T-Rex algorithm on a subset of pixels. What we do then with the list of peaks is clustering, and finally these clusters are analyzed by considering the average spectra from each cluster, and then statistical analyses are applied. So you all know the main challenges with this workflow, especially given the huge amount of data that we have. This is slow, can be biased, and most importantly, makes inter-tissue comparisons almost impossible, especially, again, at a large scale. And this is also why every group has developed their own workflow. What we propose is a solution for everybody because we want to generate clusters and molecular features in a fully automatic and data-driven way. How do we do it? So the pipeline takes all the spectra and calculates spectra to spectra similarity. So it generates a similarity matrix, in this case cosine, but we can apply any similarity measure. From these similarity matrix, regions of similarity are extracted to form clusters, group of similarity, that then we can visualize in the tissue. What we do next, so we take the spectra in each group and we reduce those spectra into one representative consensus. Consensus spectra are then used in the downstream analysis to identify the discriminative features, which can be region-specific if we look at one tissue, or can be tissue-specific if we look at multiple tissues simultaneously. And just to give you an idea of what we gain in speed, if we take our 600,000 data set, we put it in the skills and then we put it in our pipeline. Just to import the data on skills, it took us 1 day, as opposed to 1 hour with our pipeline. And if we want to look at the clusters on skills, we need to wait for 4 days. Instead, we obtain our clusters in 4 hours. Speed is not the only gain, but we also improve accuracy in the results. So if we compare our clusters with the reference anatomical regions from the Allen-Brain Atlas, which we can consider to be the ground truth for histological structures, we observe that clusters produced by the traditional workflow here on the top do not reflect the complexity of the reference regions, as opposed to what we obtain with our pipeline. And if we zoom in here in the cerebellum, we can increase the granularity of the clusters, and we see indeed that we find a distinct molecular region that is not present in skills. So what we see here on the top is the clusters using the peak list for the manual peak picking on the average spectrum. Actually, if we instead use the list from the T-Rex, we do see also here this region, but unfortunately we cannot then apply, we cannot then cluster the whole data set, so it just takes weeks. So generally what this tells us is that the limitation is not analytical, but it's indeed computational. Back to our workflow. So again, from the clusters, our pipeline generates these consensus spectra, and from those we really quickly extract the unique molecular features. And this is, first of all, important at the tissue level. It's a really big improvement compared to the traditional approach, where the statistical analyses are performed on user-selected peaks from the average spectrum. But at the cohort level, this is also very relevant because the consensus spectra allow us to easily perform intertissue analysis at clinical scale because each tissue can be reduced to its consensus representation. So we can go from 600,000 spectra down to 120 or 10 even spectra. And this slide is to show you the potential for scaling up. So we were able to analyze a cohort of 119 kidney biopsies, kidney transplant biopsies, which included 4 distinct rejection categories. And we are using a desktop workstation, which is really accessible. And it took us 2-3 working days. So 11 hours to create the IMZML files. We're talking here of 11 million pixels. And then if we look at the single slide with 17 tissue, we were able to obtain the unique features in 3 hours. So to summarize, there is a really great potential in this pipeline. We actually improved massively the speed, the accuracy, and reproducibility. So it can really enable more robust inter-tissue comparisons at the larger scale. Also important to mention that the approach is omics agnostic, so we can really put in any omics measurement, and in just one run we can analyze all these layers, so also makes it easy, the integration. With this, I'm done. Thank you for listening. Thank you also to Martin and Yassine for this great work.
Hello.
Yes.
Okay, yes, I'm back. Thanks again for the talk. So I do see Bishop Wadi also in the audience already from Alexandrov Lab as well. Yeah. Can you hear us?
Yeah, I can hear you. I can start sharing as well.
Yeah, thanks so much. Thanks again for the speaker, and yeah. Okay.
Should I start?
Yeah, you can start.
Okay. Do you see my screen? Let me put it on full screen.
Yeah, that's perfect.
Awesome. All right. Hi everyone. Thanks again for the kind invitation. I'm really happy to be back for the second edition, and really excited to see all the awesome talks and all the information. Really can't get enough of all of this. So again, happy to present an update on the projects I've been working on. So today I'm gonna present one of my projects, which I talked about a bit last year, but now it's a bit more developed, which is a spatial multiomics integration framework that we call SMScope. So this is a slide that Theo has already presented earlier, and I think it really summarizes the current focus or the progressive focus of computational developments with regards to mass spectrometry. And most of my PhD has been focused on three different aspects. The first is molecular identification, which I talked about last year with MetaSpace-ML. I also have two other projects, which is one regarding enrichment analysis. So this is something that if time allows, I can get into. But today I'm mostly going to talk about the multimodal integration aspect towards the data interpretation. So to present SMScope, so basically the idea behind this, or why do we need to do something like this, is that usually integrative methods linking metabolomics with other omics lag behind the growing metabolomics data. So we basically have much more spatial metabolomics data than we have different frameworks that kind of integrate those spatial metabolomics with other omics. We have many downstream analyses ignore spatial context. I cannot say that for the Luca's lipid brain atlas, which I still find fascinating. But apart from that, there is a lot of still many frameworks that are existing that ignores that spatial context, which leads to weaker observed correlations between metabolite abundance and gene expression. And finally, we find that most integration frameworks sometimes do handle modalities separately rather than jointly. So you don't always try to embed both modalities into one space, and that kind of limits the direct assessment of each modality's contribution to spatial domain discoveries or clusters, same as what Paula was talking about in the previous talk. So we developed SMScope, which I presented earlier. So this also happens on mouse brain, and this is basically the data set looks like you have consecutive tissues. One goes for multi-imaging to get all the metabolites, and the other adjacent section goes for molecular cartography, which is an imaging-based single molecule fish that undergoes metabolite identification from multi. We perform cell segmentation on the adjacent section. Then after a series of image spatial registration followed by pixel to cell convolution to basically try to assign the metabolite intensity to the cells, we get a paired matrix, so paired modalities of the cells from molecular cartography, where you have gene expression and metabolites on the same cells. So I just want to say that these are not same cells biologically, but these rather from adjacent sections, which is one of the limitations I will address at the end of the talk. I also aligned the cell mask to the Allen Brain Atlas, which was mentioned multiple times throughout the talks, which is a good ground truth for anatomical regions of the brain, so that I know which cells are in which region. So now we have a matrix for cells per region. And finally, I also performed some sort of niches discovery within each region. So try to subdivide those niches, those anatomical regions, into smaller niches based on their distance, solidly based on their distance, basically. And now having these aspects of like cells per regions, spatial niches, and paired modalities, we can ask various different questions. The first, most obvious one is, can we get gene metabolite correlations that are context specific? And I'll explain a bit what context specific means. We can perform region specific characterization, similar to what Luca did earlier with liposomes. And we can also do separate single cell analysis for metabolomics and transcriptomics. So this is a more of a late integration approach. So this is after doing the modality specific single cell analysis. Here I also introduced a new concept, not really new because it was published before, but relatively new applied to our data set, which is discovery of multicellular programs, which are basically another way of asking, we usually find cell type specific programs, but we don't usually see cell types shared programs. So that's what these are for. So to distinguish between cell type specific and cell type independent metabolic programs. And finally, it allows us to do a spatial multimodal integration in the true joint form, where we try to embed the cells, both modalities, into one space. So what are the key takeaways? So what I found is metabotypes. So metabotypes are the fancy names of metabolic profile-based defined cell populations, similar to how we define cell types. So these metabotypes can either be cell type specific or a mixture of different cell types. So when we try to connect the metabotypes defined by single cell metabolomics analysis to the ones that are in the brain, like excitatory neurons, oligodendrocyte, astrocytes, we see that some metabotypes, like the one here, 70% of which are oligodendrocytes, so this metabotype tend to be cell type specific, while other metabotypes seems to be all over the place. So they are kind of like heterogeneous. They are not, they are composed of multiple cell types, which you can expect, and that also means makes sense biologically, because you can have cell types that are sharing the same metabolic states, depends on energy demands, for example. The second key outcome is that if we look at regional specificity, so here this is a polygon-based figure that looks at the composition of cell types and metabotypes within each region based on the Allen Brain Atlas. And we try to quantify this. So here I'm using adjusted mutual information, which is very similar to adjusted rand index, which is a metric that is used to compare clustering to see how well they agree to each other. So if we compare cell types and metabotypes, we see that metabotypes tend to capture regional specificity better than cell types, at least in this particular data set, which was very exciting to see. And if we try to look more closely into different contexts, now that we have regions and niches, we try to check metabolite abundance. So basically, if we look at the correlations between genes and metabolites across the entire tissue, we don't get anything. So if you look here, the distribution of Spearman correlations across all cells, you tend to get very low correlations. But if you contextualize them, whether per region, anatomical region, per metabotype or cell type, you tend to see now a much stronger correlation. So the idea here is when you contextualize your analysis or a correlation analysis, some emergent connections or metabolite gene correlations emerge. So here there's an example where I'm showing cell type specific and region specific. So this is the ventral lateral part of the thalamus, and you can see here the metabolites and the gene expression that are cell type specific in that given region. So contextualization is key to show metabolite abundance correlation with gene expression. And if we look at multicellular programs, so again, as I mentioned earlier, these programs try to highlight not cell type specific patterns, but rather cell type shared patterns. So for example, if you look at this program here, MCP2, which seems to be more specific to the cortical areas and the hypothalamus, and we zoom a bit in a bit, we see that this metabolic profile seems to be very highly correlated across all the major cell types in terms of astrocytes, excitatory neurons, microglia, and oligodendrocytes. And that gives us an idea of which metabolites or which metabolic profiles seem to be cell type shared, as opposed to the ones that are cell type specific. And finally, when we look at joint integration, so trying now to look into both, basically embedding both modalities into one space, and try to find spatial domains based on that modality. And if we look at a bit on the importance of the modality, so here in this plot, each dot is the cell, and then basically colored based on importance. And the higher the importance here, so the higher closer to one, this is more important, defining the embedding of that cell. And we see that, for example, metabolites seems to be highly important in defining the cell embedding with relative to the fiber tracts, while RNA or transcriptomics seems to be better in the cortical areas or the hippocampal formation. But globally, we found that metabolomics tend to be more important in defining the embedding in a joint space than transcriptomics. So if we take this joint space and try to define spatial domains, so we're basically doing community detection, but now on a joint space accounting for both modalities, we can find that we still find the same pattern, that over almost all over the spatial domains discovered, we tend to see that the domains tend to have high importance in the metabolomics compared to the transcriptomics. So here I'm plotting the difference in the importance scores between metabolites and RNA across those different spatial domains. And again, as before, if we try to compare how well the spatial domains compare to the anatomical regions, we see that it's much better captures the anatomical regions compared to metabotypes and cell types, which tells us that metabolomics and transcriptomics can give us complementary better information than either modality alone in defining those spatial domains. This is really nice to see, but there are still some limitations in this data set. So one of the limitations, this of course is one data set with a very limited gene and metabolite coverage. So that restricts our ability to resolve various other cell types or cell states. Here is also again using consecutive rather than identical sections. So that is not the same biological cell types, but there's still some truth to it. So we basically try to interpret it with a grain of salt in that regard. And that the cell segmentation was performed basically on the lines of nuclei-based detection and transcriptional composition. So there is some limitation with cell segmentation algorithms if you don't have membrane markers. So this is also one other big limitation. There are other limitations, but for the sake of time, this is basically what I will share. I don't know how much time I have left, but I don't know, maybe.
All right, so let's check. You have 1 minute.
1 minute. So maybe I'll skip this part and then go directly, and we can discuss it later, basically. But I just want to say that as Theo said, we are now working exclusively in Metaspace and mostly on Metaspace Pro. So while Metaspace Academic, following the split, is handled by the Alexandrov team, Metaspace Pro is licensed by this new startup that I joined with Theodore and Lucas, and called MetaCloud. So now we're having very interesting new developments. Here I'm showing here the feature request page, which was recently posted. People here tend to want to have mass accuracy, and we are gonna deliver soon. So this is a sneak peek into what's coming up to Metaspace. This is just a very, very simple addition, and we're just getting started. So yeah, stay tuned to Metaspace and the developments, and hopefully, yeah, get more suggestions. Thank you again for your attention.
So much, Vishay. Thanks so much for the nice talk. And I think there will be also some question in the Q&A, so you can answer it there.
Sure.
Thanks so much again. So we move to the next speaker, which is Jonathan Zwiedler. I see him already connected. Jonathan, can you hear us?
Yes, can you hear me?
Perfect. Yeah, we do see the screen also. Yeah, wonderful.
Okay, great. So hold on, let me get this to the laser pointer. So anyway, it's great to be here. It's fun. It's hard to believe a year has passed since the last one of these. Time seems to fly. So today, we do a lot in this space, and I was trying to figure out what to talk about, and I thought I would talk somewhat about our ability to couple single cell mass spec to other approaches. And so we'll see a little bit about that. One of the fun things, this is just showing a picture of a cell, a neuron in particular. And notice, we always talk about resolution and maybe a cell is 5 microns. Okay, you look at a cell like this, what is 5 microns? It may be the cell soma, but individual synapses, individual field might be, could be hundreds of microns to millimeters. So even at a 5 micron resolution, you actually could be sampling 1,000 different cells, small pieces of some, large pieces of others. And so if you want to probe the brain, I tend to think of it in terms of space, time, and chemistry. Mass spec imaging is horrible spatial resolution. I'm sorry, even at 1 micron compared to, for example, STED and things at 50 nanometers, but it gives you chemical information. If you want time resolved information, then fluorescence has it. And so today we're going to mostly be talking about the chemical side with a little bit of spatial at the single cell level. The one thing to point out is that we use the word omics a lot, metabolomics. If you go to lipid maps, there's maybe tens of thousands of lipids in the brain. There are tens of thousands of metabolites. I asked Neil Kelleher, and there's hundreds of thousands of proteoforms. There's no single cell omics approach that's close to complete. So when we compare transcriptomics and
metabolomics, you have to understand one is getting close to complete, and that's transcriptomics, and one is a very small amount. And what you see depends on the details and what approach you use. And so this is why there can't be a single uniform platform, because you could pick different parts of this complex chemical space. And that's also why bridging these together is important. I oftentimes say this, but I thought it's always worth thinking about. In 1975, Hillenkamp showed this slide, which is 5-micron resolution using LAMMA, a different approach. It wasn't even MALDI, and you could get information. It was actually the first GCMS of a single cell of acetylcholine. And there was actually electrophoresis of a single cell. And so nothing here is new. It's just that mass spectrometers and techniques have gotten better. So it's a lot more routine and we get a lot more information. And so when people talk about the first single cell, maybe it goes back to the mid-70s. You could argue, with Sims, it's a little bit older than that. However, things have gotten a lot better and we have a lot more information. I wanted to go through two main points. One is the coupling or hyphenation, but the other is when you talk about spatial resolution, people talk about laser size. It's really LOD. And what I mean by that is if you go from 50 micron to 5 micron to, if you could, 500 nanometers, you're losing 100-fold in area. Even if you talk about single cells, you're moving, for example, 10 cubed 1,000-fold in volume each time you do that. And you can't do better than detection limit. Obviously, you can do a better probe, but just talking about detection limit, this is some data we took almost 15 years ago with an ultraflex stream on a good surface, on a clean surface with a peptide. We could routinely detect about 300 zeptomoles. Now, when we put a 300 zeptomole spot on the target and hit it with a laser, we would see, for example, this peak with mass spec imaging. Interestingly enough, if the peptide was radioactive, we actually only used about 30 zeptomoles. And so the point of this is that detection limits work if you can actually have your cells as no salts and everything worked. And so really sample handling is really important. As I mentioned, the better probe, this is SIMS. This is actually, 400 nanometer resolution. We can go to 50 nanometers. And so all of those work. For today, I'm going to talk about basically mostly physical separation, but we did do some tissue expansion. This is just one of our examples. This actually was done before, this was done by optics, so even before the hydrogel based. And this is, we just put tissue when we could only image at 50 microns, and we put beads that were 10 microns. And if the beads are separated, then we could get single cell resolution because each bead was different, and we could actually have trypsin or chymotrypsin on different beads and even do proteomics. And we can do even subcellular measurements. And so lots of tricks, and other people have done things like this. The rest of this talk, I think all of it but one slide, we're doing this, and that is we scatter cells. This is actually 10,000 cells, happens to be a tumor and a not tumor. And we can image those, and we know where we are from optics to otherwise, and we can use different probes to know which ones. And it takes 1 second per cell. If there's large empty spaces, we don't look at them. And then this is just a bunch of cells that we look at and we can optimize. One of the big advantages of this is we can over extract. We can put way too much liquid on, we can extract, break vesicles, we get really perfect detection ability in this, and yet things don't spread to the next cell because they're separated by, for example, a hydrophobic space. And so we get higher sensitivity with this, in some cases for things that are in a vesicle, maybe by an order of magnitude, others less so. So we're optimizing and each approach separately. That is the extraction and spatial resolution. And so this is again showing that last picture. We can get individual lipids. We can do 30,000, 50, 100,000. We've even done a couple 100,000 cells in this way. So let me give you some examples. One of the keys about this, though, is, and this has now become known, but we were really, 10 years ago when we published this, almost 10 years ago, people were a little surprised. This is, for example, an ultraflex stream. It's a MALDI experiment. We can then move to another lab across an 8-minute walk. And this is the same thing now on a Celerix. Same cell. We never miss a cell. This is a SIMS. So the spectra just are low molecular weight, and this is MALDI. And we can keep on measuring the same cell because we're not using everything up. One of the nice advantages of, for example, a desorption approach. This is, Peter Nemeshorty did a great job introducing this, capillary electrophoresis on a single cell after we did MALDI. And so we can get a metabolite profile and we can get a peptide profile. Now, I've shown this before. but I'm just going to show a couple older slides just because I like this example. And that is, these are individual 5-micron islet cells from a rat islet. And we know that these cells are alpha cells because they have glucagon, and these are the metabolites. And we can even say that these cells have dopamine in them. And you can see that because there's dopamine, we can do standard addition. We can quantify how much dopamine is there. And what's interesting about this is that actually wasn't known before we did this, even though the enzyme to make dopamine had been shown to be in alpha cells. And so you get some fun information. We can do this with human cells. These are living human cells. And by the way, people talk about long distance measurements. We sometimes take measurements from Antarctica and the samples are shipped here. In this particular case, living islets from a human islet transplant center were teleported FedEx to us. And then we dispersed them and did measurements. These are individual cells from one human donor, and different islets have different amounts of somatostatin or different number of cells that are alpha cells with glucagon or insulin. And we can even start looking at unusual cases, in this case in type 2 diabetes. And I show you this. This is the processing of the glucagon into, for example, glucagon, oxyntomodulin, GLP-1. And all of these we see in islets, not in gut cells. Islets specifically that have, from a human donor that have type 2 diabetes. And here is just an example of lots of the cells in this particular islet have insulin, but one cell has oxyntomodulin. And we were able to identify it. There's the multispectra. And I still like this example, even though we have others, because people talk about measuring subtle differences in the lipid or not. In this particular case, this cell has completely given up on making glucagon and is making oxyntomodulin. The change in signal-noise ratio is a couple thousand to 1. So the key, though, is that you can't do 100 cells. You have to do tens of thousands of cells to find these. Here's some new data, and that is we wanted to look at this, oxyntomodulin and GLP-1, in the gut. The problem is it's 1 in 100,000 cells in the gut make this. And it turns out when we did mass spec imaging, it was really hard because these signals were swamped by all the common materials. And it was even worse than that. And that is some of the, when you slice, you don't get the same amount each time. And so what we did is we labeled the cells, isolated them by hand, or in this case, by FACS sorting. And when we did that, we actually discovered that some cells not only are GLP-1 cells, for example, that would be an L cell, they also are K and I cells or L cells. And so they're all mixed up. There's almost a combinatorial mix-up of what these enteroendocrine cells. Now, in last slide was a human. This is going back because what we're doing is shows a picture of it. It's a mouse. And so, okay, I'm going back and forth between models, but you can find exceedingly rare cells and try to probe them with these approaches. What are the cells? Now I'm going to jump to a brain. So I apologize for the number, but this case, what we did is we used the Ambergen approach where we did mass spec. And then I'm actually showing you staining with fluorescence. But what we did is we used mass tags, and we can then say that this particular lipid profile goes to an astrocyte because it has GFAP, or a neuron because it has neurofilament, or it has a oligodendrocyte. And we can then start seeing lipid differences by cell type. This complements what we heard earlier today by zip codes. We can absolutely assign cell types, and some of the lipid differences are quite large. We can actually be subtle and look at differences between activation and synapse formation because there's certain tags for that, and we can actually start looking at subtle differences. Now, when I showed this to some colleagues, they say, that's great, but I don't want mass tags. I don't want immunohistochemistry. I want transcriptomics. And so this is some work that we did with Jim Eberwein, where we did single cell lipidomics, and then we teleported our cells to Jim Eberwein at the University of Pennsylvania. Now, if you don't know the name Jim Eberwein, that's fine, but he is the person who invented the entire field of single cell transcriptomics. He published the first ever single cell transcriptomes in 1991. And so long before there was anything commercial, and he still pushes the field. And he told me we were doing everything wrong because we weren't adding RNases. And what we did is we just basically took the cells, we optimized it for MALDI, and we took the hit on transcriptomics because we figured he could handle that. And what we get is we can now, in this particular paper, we get, actually Jim gets, instead of about 15 to 20,000 transcripts per cell, he's getting about 8 to 10,000 because we messed up his measurements by adding MALDI matrix first and all that. But we can do single cell transcriptomics after we did MALDI. Obviously, you have to move the matrix and things. And so this work has just been, so it's not an adjacent slice. It's not the Allen-Brain Atlas. And so, yes, you can do a lot of different things on the same cell. this was really fun. Work with Neil Kelleher all the time, and he says metabolites, I'm going to get yelled at when I say this, are not where it's at. We need to be at proteoforms. That is not even proteins, but proteoforms. And so we started working with him, and here's some work that he did based on some work, Julia Laskin, which we published last year, where we did single ion mass spec. And you can see this cell is about to eat, this drop is eating a cell, and we're extracting, and we do a cellogram. And earlier this year, this was really fun. In the summer, we had a paper in Nature Biotechnology. I say we because we're the ones that got them the cells. You can see a couple people from my group, mostly though, this is Kelleher work. And they're able to do, in this particular case, about 10,000 individual cells. They get very little information per protein. 1 in 1,000 proteins actually are ionized into their system, but they're doing single ion mass spec, and so they can get the charge distribution and a lot of information from every single ion that they happen to measure. And we're able to then get a lot of information on individual cells, but this is a metabolomics. And so what we do is you can isolate the cells in Illinois, at University of Illinois, teleport them up to Northwestern. They make this measurements, and they teleport them back down to us. Boy, there's a lot of FedEx involved. And this is our first example now, just very recently, where we're looking at where they scan the area, where they didn't scan the area, and we're actually able to get a little bit of the cell goo that's left over. We're able to actually still do the lipids. And so their extraction does the proteins. That's what it's optimized for. And whatever's left, we can measure. And there is a change. And so quantitation is going to be a little bit hard here because we sort of messed up the measure. Oh, no, the Kelleher group messed up the measurement to optimize theirs. Now, I would actually say that's okay because the proteoforms is the hardest measurement to make. Just showing you because there was a couple we can do single vesicles. I showed this last year, so I'll go really small. These are 500 micron to 500 nanometer, 1 micron vesicles. We can separate them well, even though our laser spot is closer to 5 microns. We're doing 500 nanometer objects. Here's lipids and here's peptides in these dense core vesicles, and we can actually get information about differences in vesicles that have different cargos. And I thought I would show you a last slide. It's only one last data slide. What this is doing is taking single mouse, in this particular, rat organelles, in this case, the mitochondria. So we saw some wonderful differences in lipids, and they were able to get this great data by really, really intelligent and fancy probes. In this particular case, we're just isolating these mitochondria from different organs, and then we can get some of the lipids in these mitochondria. And one of the reasons to do this is Jim Eberwein with transcriptomics has shown that some metabolic diseases, if all the mitochondria have a problem in transcriptomics, it's called death. But if 1 or 2% have a problem in the transcriptomes, the mitochondria are different, and then you end up actually having certain types of disease states. And so the heterogeneity in the mitochondria of the individual cell completely correlated with disease state. What's different? And there's evidence that it's even how the mitochondria fizz and fuse. And so we would think that was at least correlated with lipids. And so we are trying to look at that. And this is just the first example where we are looking at that. I'm not showing it, but we actually see a lot of heterogeneity in mitochondria in the brain, and surprisingly less heterogeneity in the heart and the kidney, probably by cell types. And these are just some of the lipids that we see. So this is pushing the limits of detection for an organelle, at least in our hands. This is about the smallest things we were able to do. You can see we've spaced them very far apart. This looks like microns, even though we can visualize them, and then we can see these. And our laser is much bigger than the mitochondria, but again, this works because of the way we've separated out the mitochondria. Next step is to do this within individual cells, and that takes a lot higher, more sampling steps because we're trying to then to look at mitochondria within an individual cell. I forgot about this. The last bit of data is this is some work. This was published, by the way, in Nature Communication. And if you look, it was just yesterday. So I thought I would throw this on. This is with a postdoc, Tim Trinklin, and some collaborators. And we're doing, in this case, mass spec imaging of an Alzheimer's model, in this case, a 5-FAD. But the reason I decided to throw it in is you can look at plaques. And we, our collaborator, Orly, looked at the 5-FAD. We did some other models because at this time frame is the plaques are just starting, and they're sort of on the scale of individual cells. So they don't have to be cells, right? They're well spaced, and we could actually then do our mass spec approach where we actually have the tissue on there, and we can just look at what's happening in and around plaques. And we can then look at lipids that are on the plaque or plaque localized lipids by just getting a little array around each one. And we can see plaque enriched indepleted lipids by treating these hotspots, sort of the same way we're doing. It's a very different experiment in terms of its tissue, but we could actually then localize where these are and actually see not only are the lipids plaque enriched and plaque depleted, but they actually also are brain regions specific, which makes sense because as we heard about zip codes, again, if the lipids are different in different brain regions, then it's not surprising that the plaque-associated lipids are, but there are brain regions that seem to be much more resistant to forming plaques. And is that related to the lipid changes in those regions? And this is the type of thing that we can study, all because of this ability to basically localize and measure these individual units. Final conclusion is just simply, our measurements are trading off chemical detail, spatial resolution, throughput, and we're combining single cell and mass spec with a lot of different modalities, often on the same cell, once in a while on addition slices. And it's that combination that gives us really unmatched types of chemical information. And then the last thing I really do want to say is that when I talk to my biological collaborators, oftentimes they act like the list of molecules is known. But every time we start doing this with new tools, we keep on finding unexpected and novel molecules. And the last thing I want to say is we always want to go small, but I'm finding the hardest thing to do is actually go small and large. Some cells in a mouse go from the brain to the toe of a mouse. How do you deal with things that are centimeters long? That the active area is still in the nanoscale. And that type of dynamic range in terms of chemistry, volume, and time are really difficult to deal with. And with that, I think I'm truly out of time. And so I thank you very much. Any questions?
Yeah. Alessandro, can you jump in or read the questions?
Say that again?
No, I was actually asking Alessandro to come in and, yeah. Xinji, can you go on?
Hi. Okay. Do we have any question for this talk?
I think we're out of time.
Okay, we are off time. Yeah, we have a problem of the next speaker because we cannot give her the access to this conference room. So I don't know. Can I ask kindly for the next speakers, Rikintel, to be the next speaker? Hello, Riken. Hello, Riken. Are you there?
Yeah, I'm here. Am I the next speaker?
Yeah, can.
Sorry,
Yeah,
it was
much.
a bit
Thank you.
unexpected. Let's see. Okay, give me a time to figure this out. Is this sharing the right screen?
Yes, we saw the screen.
Okay.
Thank you so much.
Me see. Okay, hi, my name is Reka. Thank you for the opportunity to speak in this session. I'm speaking on behalf of my PhD advisor, Jeremy Baskin. I'm currently a postdoc in Alice Tin's lab at Stanford University, and today I'm excited to share my PhD work. Okay. So first, this is the textbook depiction of cell and organelle membranes, but as you know, this is an oversimplified view. Because each cell and organelle is surrounded by lipid bilayers, and each membrane has a unique composition of lipids. And I don't need to emphasize to this audience how spatially heterogeneous these membrane structures are, and Professor Nadler just gave a beautiful introduction on how cells maintain this heterogeneity. But here I want to highlight the importance of membrane-associated proteins, which account for roughly 50% of the membrane mass. At each membrane compartment, lipids and proteins form a dynamic and complex network. There are membrane-embedded proteins that cluster specific lipids, lipid transporters that move lipids from one membrane to another, and lipid metabolizing enzymes that convert one lipid into another. There are also lipid-binding proteins whose localization and function are regulated by lipids. Our goal is to decode lipid-protein networks at subcellular or even at suborganelle levels so that we can address key questions in membrane biology. Where lipids are made and where they go,
how these localizations are established, and what biological functions they serve. The Jeremy Baskin lab at Cornell University develops molecular tools to address these questions. The group is focused on 3 project themes. First, imaging lipid biosynthesis, signaling, and interorganelle transport using bioorthogonal chemistry. And second, developing membrane editors whose activity can be controlled by light in order to modulate organelle lipid composition with spatial temporal resolution. And third, applying these tools to advance our understanding of lipid biology. And in today's talk, I'm going to discuss our most recent work, which involves applying membrane editors in combination with proximity labeling in order to map lipid protein networks in cells. This work uses one of our recently developed membrane editors called SuperPLD. PLD is an enzyme. Its full name is phospholipase D, and it produces phosphatidic acid, which is also called PA. PA is an important signaling lipid that have multiple functions in cells. I'm going to skip the details of our engineering efforts because they have been already published here. But long story short, SuperPLD was developed from a bacterial PLD that catalyzes the same reaction as mammalian PLD. And this SuperPLD is expressed in the cytosol and recruited onto a membrane of interest using a system called OptoPLD system, which is based on light-induced heterodimerization between Cry2 and CIBN domains. And this membrane-recruited SuperPLD can produce PA at the site where it got recruited to. And to capture the proteomics changes in the membrane environment, we used a proximity labeling enzyme that can label nearby proteins through a promiscuous reaction. This proximity labeling enzyme is genetically fused to an organelle targeting tag for its expression on the same membrane targeted by OptoPLD. So to summarize the concept, here SuperPLD is used to feed the PA molecules on the membrane of interest, and the proximity labeling enzyme is used to fish out proteins associated with PA-fed membranes. So we call this concept feeding fishing approach. Our first step was to choose which proximity labeling system to use, because there are 2 widely used proximity labeling enzymes that rely on different chemistry. One is called Apex and the other is called TurboID. Both enzymes tag nearby proteins with biotin, and the biotin-related proteins can then be isolated by streptavidin pull-down and identified by Western blotting or mass spectrometry. We compared Apex versus TurboID for their ability to selectively label the membrane-associated proteins. And to test this, we tethered Apex to the plasma membrane and recruited a model protein, in this case OptoPLD, to either plasma membrane or lysosome. Oh, no, plasma membrane or ER. And if there was membrane selective labeling happening, we would expect to see more labeling on the plasma membrane recruited OptoPLD. But this was not the case. By contrast, plasma membrane tethered TurboID labeled plasma membrane recruited OptoPLD much more efficiently than the ER recruited OptoPLD, and similar selectivity was observed for ER tethered TurboID. So, sorry. Sorry, I got distracted by the chat. So from this comparison study, we concluded that TurboID has better membrane selectivity than Apex. So we decided to choose it for our feeding fishing proteomics. We used lentiviral transduction to express these necessary components for the feeding fishing proteomics in hex cells. And for the target organelle membranes, we chose plasma membrane and lysosomes because these 2 are the key locations that are known to be important for PA functions. To perform quantitative proteomics, we included a catalytically dead PLD that cannot produce any PA as a negative control, So this allows us to compare the ratio of proteins labeled by turbo ID on PA-fed versus PA-unfed membranes. And these volcano plots show the proteins that are either enriched or depleted on PA-fed plasma membranes or lysosomes. We identified a number of proteins that are known to be associated with lipid transport and metabolism. Among the proteins that are more enriched on PA-fed membranes, we found lipins, which degrade PA to diacylglycerol. And among the ones that are more depleted on PA-fed membranes, we saw diacylglycerol kinase, which can produce PA from diacylglycerol. So this indicates that on PA-fed membranes, there are more proteins that can degrade PA, and there are less proteins that can produce PA. So these are really satisfying to see because they gave us confidence that our feeding fishing approach is working. Among the lipid transport-related proteins, we detected PATP-NM1, which is also called NER2. And this is actually a reported PA transporter that's known to transfer PA from the plasma membrane to the ER. And to further study the functional relevance of this NER2 and as well as other lipid transfer proteins that are highly enriched in our feeding fishing proteomics, we did the follow-up studies. For this, we did lipodomics in HEK cells expressing super PLD used to feed PA onto either the plasma membrane or lysosomes. Selected lipid transfer proteins were co-expressed in the same cells, and those proteins were NER2, PDZD8, ORP1L, or STP2, and all of these proteins expressed well in HEK cells. Interestingly, expression of these lipid transfer proteins caused marked changes in cellular levels, not only of PA, but also of other phospholipids. And this happened when PA was made on plasma membrane, as well as when it's made on the lysosomes. Expression of NER2 or SCP2 was particularly effective in reducing global PA levels. And on the other hand, ORP1L expression especially when PA was fed on lysosomes, led to unexpected increase of PA levels, which was quite interesting to us. And there are so many things we can talk about from these results, but for the sake of time today, I want to focus on NER2 and STP2. Because their expression showed a clear reduction in global PA levels, we also investigated whether this effect occurs at local subcellular levels. To monitor local PA metabolism, we used a PA binding probe that was made by fusing GFP to a PA binding domain from SPO20 protein. This probe localizes in the nucleus in the basal state, but it gets recruited onto membranes by sensing PA accumulation. So this can be used as a sensor to monitor PA localization. When super PLD was recruited on the plasma membrane or lysosomes to activate PA production, we could see partial translocation of this probe to each organelle. And this is in the control cells, but in the cells of overexpressing NER2, this local PA enrichment was largely abolished. A similar effect was observed in cells expressing SCP2, and shown here is the quantification. So these lipid transfer proteins that were identified in feeding fishing proteomics can indeed promote PA metabolism in cells by reducing local PA enrichment. And so higher expression leads to better PA clearance. But is the opposite also true, meaning that if these proteins are depleted from the cells, would we see more local PA enrichment? To test this, we used siRNA to deplete endogenous NER2, and in these knockdown cells, PA production seems to cause greater cell toxicity, potentially due to compromised lipid metabolism. So to mitigate this effect, we switched from super PLD to lab PLD, which is our more recent version of membrane editor that has a lower background toxicity. And just like in the previous experiment, we could see PA enrichment on the lysosomes when the PA was made on the lysosomes. But what happened in NER2 knockdown cells was completely unexpected, because instead of seeing PA enrichment on lysosomes, in the knockdown cells, we saw PA enrichment on this perinuclear structure that colocalized with Golgi. This unique pattern was also seen when PA was fed onto the plasma membrane. NER2 is canonically reported to transport PA from the plasma membrane to the ER, where PA is converted to CDP-DAG and eventually to PI. However, our results indicate that in NER2 knockdown cells, lipid metabolism might be rewired to compensate for the loss of NER2, and that the Golgi is somehow involved in this alternative PA metabolism pathway. And this probably reflects the fact that PA is an essential signaling lipid whose levels should be tightly regulated in cells. And this leads to the last point I want to discuss, which is how remarkable cells are in maintaining PA homeostasis. In our lipodomics analysis, super-PLD was expressed to feed PA on either the plasma membrane or lysosomes, and blue light illumination was used to activate super-PLD, and this resulted in an acute increase of PA levels. So this is looking at kind of short-term effect and of PA production, and the increase was more pronounced when PA was made on the plasma membrane than when it was made on the lysosomes, which indicates slower turnover of PA on plasma membrane. And again, this change reflects the short-term effect. And we also compared PA levels between these two conditions, where the cells are expressing dead PLD versus when in the cells expressing super PLD, because we expected to see an even larger difference between these two comparisons due to the PA accumulation happening from the background activity of super PLD. However, what was, well, quite unexpectedly in super PLD expressing cells, we actually showed similar or sometimes even lower levels of PA levels than in cells expressing dead PLD. So this was quite puzzling, because super PLD should have made tons of PA, and where did these PA molecules go? We then checked levels of other phospholipids and observed this massive accumulation of phosphatidylglycerol, PG. And this suggests that cells can remove excess PA by converting PG to converting PA to PG via the CDP-DAG pathway. And moreover, considering that super PLD uses PC as a substrate to produce PA, and yet there was no marked reduction in PC levels, suggests that PA can also be, a significant portion of PA can also be converted back to PC to replenish the PC levels. And this should happen via Kennedy pathway. And to further investigate this massive PG accumulation happening upon PA production, we tested another experiment. And this is the final data that I'm going to show. The PA to PG conversion is reported to occur in mitochondria, with the first step mediated by CDP-DAG synthase that converts PA to CDP-DAG. And interestingly, we found that knocking down the mitochondrial CDP-DAG synthase, called TAM41, largely abolished the PG accumulation that was observed in super PLD expressing cells. So this supports the hypothesis that PA, whether it's made on the plasma membrane or the lysosomes, can be trafficked to mitochondria by lipid transfer proteins, where it is eventually converted to PG. So in conclusion, we developed feeding fishing proteomics to identify proteins involved in lipid metabolism. And our proteomics and lipidomics studies revealed diverse strategies and pathways that cells employ to maintain tight PA homeostasis. Looking ahead, we aim to expand this feeding fishing approach to study regulatory mechanisms associated with other lipids. And because many of these mechanisms appear to occur at distant membranes mediated by cross-membrane lipid transport, it would be interesting to perform feeding fishing studies on different membranes rather than the same membranes. And the study I discussed today is available as a preprint, but we added a lot of data during the revision process, and this final version will be published very soon, so please stay tuned. And with that, I'd like to thank Jeremy and the Baskin Lab, and thank you so much for listening. I'm sorry I went over time. Thank you.
Okay, so thanks so much for the nice talk. So the next speaker of today is Shane Ellis. And I see that Shane is already here. No, actually, I think that now we can, okay, because there is actually a question already in the chat that I can maybe ask you before going to Shane directly. So all the reports by Tsima localized near to at ER Golgi, MCS, and would your data support at least functionally the localization?
Oh yeah, I think I know the study here. You're talking about the NER2. Wait a minute, I think so. It's kind of like quite complicated to explain. So in their report, they observed that NER2 knockdown. Wait a minute. Sorry, sorry. I'm kind of. Our results do align with their report, and one hypothesis is that upon in NER2 knockdown condition, cells try to replenish the PC levels via Kennedy pathway by increasing the PA flux that goes to Golgi. But sorry, there's like, can I try to answer by chat later, because I'm kind of
For sure. You
I
can
don't think I have capacity to fully answer it right now.
Don't worry, it's fine. You can also ask by chat. Actually, we can also move to the next speaker then that is already here. Thanks so much for the talk. Thanks.
Thank you so much.
Thanks. So we can move. Shane Ellis is here. Do you hear us?
Yes, I can hear you. Can you hear me?
Yeah, perfectly. Thanks. Can share the screen. Can you?
I think it's good afternoon for most of you. Yeah, I'll share my screen.
Yeah. Much for being here this early for you. Yeah.
Alrighty. Let me just. Sorry. Is that working? Can you see it?
Yeah, perfectly.
Okay. Yeah. Well, thanks for putting together such a really interesting online symposium. Yeah, it looks great. And thanks for inviting me. So yeah, I'm going to talk a little bit about some of the aspects we've been working on in developing new methodologies to enhance spatial lipidomics. We also do some non-lipidomic stuff, but mostly focusing on lipidomics. So yeah, we're in Wollongong, which is just a little bit south of Sydney. It's a little coastal town, but it's a really nice place. The university is about here. Kind of our research, yeah, we do a lot of mass spec imaging, lipidomics, and we're kind of focusing along three main themes. One being kind of instrument development, so developing post-ionization methods and ultra-high resolution imaging methods, which I'll talk a little bit about today. Method developments as well, around derivatization, isomer resolve, lipidomics, quantitative imaging, and also various applications in neurodegeneration, cancer, and also single cell analysis as well. But what I want to talk about today is a little bit on the quantitative imaging and how we're using this in a couple of different projects. and also some of the ultra high resolution methods we've been working on. So firstly, one of the challenges we set out to address for a few years ago now is kind of how can we really get quantitative lipidomics imaging data, and doing this in a way that we can do this on a large scale and across many different lipid species. So obviously there's quantitative imaging where you have internal standards, but the drawback of that is the internal standard only allows you to quantify what essentially the same class, or in the case of lipids, the same lipid class. PC internal standard quantifies PC lipids. But we wanted to do this on a more holistic scale. So we actually worked on, with Avanti, this internal standard mixture with optimized concentrations where we can quantify 13 different lipid classes. And the idea is pretty simple. You take this mixture, you can spray it over, well, it's optimized for brain tissue, but it works on many other tissues. And then in the end, you can quantify all these different classes, and I'll explain a little bit how that's done. As part of the original study, we also looked at different instruments, the effect of mass resolution, and the ability or inability to resolve different isobaric interferences, and what effect that has on quantitative accuracy as well. Obviously here, the general take-home message is higher the mass resolution, there's less interferences, and it's generally a little bit more accurate. But the overall result is you get images across all these different lipid classes and all the species within those classes where you can express them as concentration, absolute concentration, so picomole per millimeter squared in our case. So the idea in doing this is you do your imaging, you spray your standard, you do your imaging experiment. We do this in both negative ion mode with MALDI and positive ion mode with MALDI2, especially because that allows us access to a lot of these glycosphingolipids that we don't detect particularly well with conventional MALDI. And then we have a data processing workflow whereby every single peak in the MSI data set, we have to assign it to a lipid class based on accurate mass or other information. And then once we can assign it to a class, we know what internal standard peak it has to be normalized to. And we do this on a per pixel level because different lipid classes need to be normalized to their own respective internal standards. And this is just some of the data we acquired on mouse brain using this approach. And you can see overall, it's kind of reassuring that the internal standard normalized images generally look the same as what you would normally get by tick normalization or even in the absence of normalization. So it's a per pixel division of this image divided by this image. And you can already see why we need this, because we get different ionization efficiencies across the tissue. So with these reference images, which are the internal standards, you can notice two things. One is there's much higher signal off the tissue because there's much less ion suppression. And two, even though it's sprayed homogeneously across the tissue, across different parts of the tissue, the signal's different. And this is exactly the effect we're trying to correct for by doing the internal standard normalization. So here you can see just some examples of lipids as M-H ions, and you can already see some subtle differences. So here, for example, with this PI 38.4, we kind of look like there's a depletion in the hindbrain, but if you do the tick normalization and correct for a lot of these region-specific effects, you can actually see the concentration in the hindbrain is kind of underrepresented when you don't do the internal standard normalization. So you're comparing this region here to this. One of the other benefits we find with this is, especially when we do MALDI2, is sometimes you get these kind of striping artifacts, maybe because of a big matrix crystal that's sitting on the tissue. So this happens sometimes, but when we do the tick normalization, it corrects for all of these as well, which is kind of nice. So in the end, what we can do our imaging experiment, we can do the post-MSI histology, and here we just, crudely define 5 different regions. This was done together with Aspect Analytics, and then from each region you can extract out the quantified lipid profile across all of the classes. So here we've just done the 5 different brain regions, and you can already see that this is working as what one would hope. So for those that do a lot of lipid imaging on mouse brain, or brain in general, your highest signals are often the PIs in the gray matter or the sulfatides in the white matter. So these are usually by far the highest signals. But you can actually see if you do the internal standard correction for the quantification, PEs and PSs are actually present at higher concentrations than the PIs and the sulfatides. So they just suffer from more ion suppression. And this reflects the concentrations you would get if you did LCMS or quantitative shotgun lipidomics. So this is, well, we think this is really great because now we're getting kind of a much more accurate reflection of the lipid profiles and the concentration of different lipid classes relative to each other. So it's really reflecting the true lipid composition of the different regions. Obviously, we can do this in positive mode too, so we can look at the different lipids, the hexosyl ceramides, sphingomyelins. PCs are especially a little bit tricky because you have the overlapping plus H and plus Na isobaric interferences. And in this case, we couldn't use the potassium adduct of the internal standard because it overlapped with another signal that would pop up, I think a 13C isotope or something. So what we actually decided to do here was we actually split whether we used the plus H or the plus sodium adduct. So generally for the shorter, less PC lipids with, say, less than 2 or 3 double bonds, we use the plus H species. And for those that have more double bonds, we use the plus sodium. And this gets rid of a lot of that interference as well. So one area we're applying this now is looking at Parkinson's disease, both in mouse and also in human tissue, both on the neutral glycosphingolipid level and also the ganglioside level. So our motivation for this is partly that one of the primary risk factors of developing Parkinson's is mutation in the GBA gene, which inhibits the degradation of glucosyl ceramide and glucosyl sphingosine. So because this is not degrading anymore, you can get a shift in sphingolipid metabolism because you're not breaking these down, and these can go into other pathways, particularly into the gangliosides. So firstly, we wanted to just explore how we can, how accurately can we use mouse tissue to reflect disease in human brain, and especially what are the differences in ganglioside composition across mouse brain and human brain. So in this first small study, we take mouse brain and we take 3 human brain sections, frontal cortex and midbrain. And here we've taken the same internal standard approach, but now we're doing it for ganglioside. So we have 3 different ganglioside internal standards that we spray onto the tissue at an optimized concentration. So this allows us to normalize, obviously, these endogenous ganglioside classes, but we also kind of use our best guess and guess which ones of these would be most reflective of the ionization of other ganglioside species as well. So for example, the GT species, we normalize back to the GD species. The workflow is pretty similar. We spray this on, we put matrix. We're doing this on an Orbitrap fusion with a MassTech multi-source. Then for the mouse samples, we can do the AMBER registration and automatically pull out all the regions. And then on the human samples and on the mouse, we also get pathology annotations as well. And then it's the same workflow. We can do the quantitative ganglioside imaging and profiling within all of these different defined regions. So firstly, just looking at the mouse brain data. So we register it to AMBER, we do the Luxol fast blue staining, and we can get these 12 different regions. I'll say one of the other benefits we get here is for those that have done a lot of ganglioside work is internal in-source fragmentation can be quite challenging, so especially loss of the sialic acids. So this means GD, gangliosides, can also fragment back to GM when they lose a sialic acid, but GM is a real endogenous ganglioside as well. But with this internal standard mixture, we can actually correct for that. So the GD fragmentation is automatically corrected for by the GD internal standard, but the GD fragmentation leads to an overrepresentation of GM fragmentation, of GM signal. So what we can do is we take the GD internal standard signal, we can look at in every pixel how much the in-source fragmentation has occurred, because we can see the corresponding fragment. Then we can do a per pixel correction of the in-source fragmentation and essentially subtracting away the signal of GM that's coming from in-source fragmentation. So what this means is you get a more accurate representation, and you can kind of see this here if we just look at the class level comparison across the different regions, is if we don't do this in-source fragmentation correction, we get more GM signal than we get for GD but there's more GD in brain, so we expect to have the GD signal higher. But if after we do the correction, we get kind of the correct ratio, and we're correctly seeing that there's more GD than GM. And of course, we get all these quantitative values. This is the summed signal across all the species that belong to all these classes. Then, of course, we can do the breakdown per species. So in this case here, these are the different GD1 species, and we can track their relative concentrations across the different brain regions. Obviously, overall, kind of expected, we see more ganglioside signal in the grey matter, which is probably reflected of their high concentration in neurons. So then we've looked at, I guess, how does the mouse brain differ from the human brain? So again, looking at human frontal cortex, human midbrain, and what we compare here is firstly just white and grey matter regions from these regions. So from the mouse cortex, we compare white and grey matter. From the human frontal cortex, we compare white and grey matter. The midbrain samples we have are mostly white matter, so that's why I don't show them in here, but we do have them. So of course you get the quantitative images across all the different regions. You can already see there's some overall differences. There's overall less signal in the midbrain, mostly because it's mostly made up of white matter, at least in the samples we received. But what we also notice is that generally the human favors longer chain ganglioside species. So you can kind of see that here, and if we compare the mouse and the human, we see more of the 36-1 species in the mouse, which is the D18-1 sphingosine chain. And then in the human samples, we see more of the 38-1 species. And this mostly consists of the D21 chain, sphingosine chain, which we can tell from MSMS. You can kind of see that more reflected down here as well. So the humans, we have more, obviously more in the grey matter, but more of these longer chain 38-1, 48-1, 42 species than we see in the corresponding mouse. We can also take all these regions and just look at all of the data and see kind of which ones are most significantly upregulated in the mouse and in the human. And again, you can kind of see the same trend. These longer chain species across all the different classes, mostly GD, GT, GM, because these are the ones we get the most signal for. Yeah, we see more of these in the human. So what you see here is yellow is the fold change for that species, human versus mouse, and then you have the corresponding p-values as well. So you can see, yeah, and then these shorter chain species that are favored in the mouse. So this was just a proof of concept study we've been doing, just firstly to look at human versus mouse, but also to develop workflows, because we have a larger study that we've just finished acquiring all the data for, where we have 45 human cortex and midbrain samples, both from Parkinson's and controls. And we've run these across two different workflows. One is this quantitative ganglioside workflow, and one is using the splash mix on an instrument I'll explain in a minute, where we get all the a lot of the phospholipids and neutral glycosphingolipids quantified as well. We're in the process now of analyzing all of this data, but the initial results do look quite interesting. So I'm going to shift gears a bit now for the second half and talk about some of the more instrumentation projects we've been working on. So we started we do a lot of post-ionization work, so both with MALDI-2 and now also using plasma post-ionization. So a few years ago, we started a project to explore post-ionization on a timsTOF instrument using this Secret, the plasma ionization source from Plasmion. And the benefit was, the idea was, can we get the benefits of post-ionization without having to worry so much about fine laser alignment and sensitivities around that. So the idea here is we took a timsTOF Pro, and we essentially have the stage and the laser from the Flex mounted externally. We have a laser, it hits the sample. The sample is still coated in matrix, and this is really just to allow us to get broad desorption of species across from the surface. It's not so much involved in the ionization so much. Everything gets sucked in through this capillary just purely by the vacuum and into the secret source, and then things ionize, and then we can detect them. There's some intricacies to this that we still don't fully understand. So different lipids or different analytes have very different temperature dependencies for the inlet capillary. So there's some very interesting desolvation type processes happening as well. But to our surprise, this worked very, very well from the very first time we tried it. So this was kind of some of the first data we took. This was published a few years ago, but you can see this works really well. This is mouse brain, 20 micron pixel size. This is a representative single pixel spectrum. We have a dynamic range of almost 3 orders of magnitude. And what's really nice is kind of this profile very closely resembles what you'd see from a good MALDI-2 spectrum. And by that, I mean they're big peaks now. This is positive moded, not PCs, but these are things like PEs, hexosyl ceramides. We see the lactosyl ceramides, and even species we don't normally see in positive ion mode, so things like protonated PSs. These all pop up. We can confirm all these with MSMS, these ceramide phosphates. So we're getting very, very rich data that very closely resembles MALDI-2. We've obviously used this now on a lot of different samples, both tissues and even single cells. There is some fragmentation, obviously, but it's not so bad, and it's surprisingly only really confined to PC lipids. So a lot of PEs, the hexosyl ceramides, PSs, don't really fragment much at all, but we don't see we see very little PC signal. Most of that goes to these head group loss fragments. But because these mostly come from PC or tags, if they're present, we can kind of use these as a readout for the PCs themselves. We also see the demethylated fragment, so the M-CH3 fragment from the PCs as well. This obviously overlaps with PEs, but these you can separate with the TIMs. So because this worked well, we then decided to see how far we could push this in spatial resolution. So we built this into a transmission mode source, and by that, so for those not familiar, what I mean is transmission mode means we bring the laser in through the back of the sample. This means we can use a high numerical aperture objective right up against the back of the slide, so short focal length, and this means we can focus the laser down much, much tighter. But then the overall setup's pretty similar. It's just laser comes in through the back, and then you can see it's the sample stage. We've got a different stage now, it's piezo, so we can do much smaller steps. And then this is just shot straight into this heated inlet capillary. Now it's a straight capillary and then into the instrument. Because we have this objective here, this also functions as an in-source 50x microscope. So this is the optical images we see in real time. So this is from an ablation array where we do a 5 micron step, and you can see we're getting ablation craters here about 1 micron in size. So we can use as a fluorescence microscope as well. So we have the different excitations, and we can use the different filters to do fluorescence microscopy in the source as well, which is particularly nice for some of the single cell work. So it's kind of similar to the Flex system that Munster have developed for MALDI-2, but now we're using it for the plasma ionization. We also developed a different way of preparing the samples for plasma ionization, because the way what's on the sample matrix, it doesn't have to ionize the material anymore. It doesn't have to function well as a matrix in the sense that it ionizes. We just need a way to shoot stuff off the surface sufficiently and capture it into the inlet capillary. So this was the first, some of the first trials we did. So this is a mouse brain sample coated in 2,5-DHA. With DHA, we struggle to get down to 1 micron craters for some morphology reasons, but we're getting 1.5, 2 micron craters. You can see it kind of works, but the images are a little bit patchy. So then what Ruben developed was this way of doing pre-staining, where he adapted Crescel violet staining procedures to remove a lot of the permeabilization steps, so we still preserve the lipids, and we essentially pre-stained the tissue with Crescel violet. And this is now the pre-MSI optical image of the sample. So it's pre-stained now. So this obviously means we can get good morphology information prior to any damage that might come from the MSI. And the idea here is we're kind of embedding and absorbing material into the tissue that helps us to sample the tissue much more efficiently. So what we see is if we do just the matrix, there's still a lot of tissue left behind after the experiment, but by having this absorber in the tissue for the plasma ionization especially, we actually shoot out the entire volume of tissue. So we're sampling not just the analytes that get extracted into the matrix layer as you would in a normal MALDI experiment, but we're sampling the entire tissue volume. And because we don't have to ionize it during the desorption event, the ionization is taken care of down the track in the plasma source. And you can see here the difference is quite stark. So this is the conventional matrix only. Then if we do the pre-staining, this is a single pixel spectrum from now what's only less than 1 micron ablated area. So we get ablated areas now about 700 nanometers in diameter. So we're now pushing down towards submicron, and we're getting very, very rich data. So you can see here, these are just an overlay of 5 different species. I'll also point out that one of the other benefits of this staining is we actually detect nucleotides as well, either as their free form or as covalent derivatized adducts, where the crescel violet has essentially in situ derivatized them. So here you can see in blue, these are the purines that we can localize here into these cells within the hippocampus. Now, these are just some further examples. So this is mouse cerebellum pre-staining. This is a single pixel spectrum. And you can really start to see that we're resolving a lot of really, really fine cellular level detail within the tissue. So there's this layer of cholesterol here that's only maybe 10 microns wide, between these two regions. And yeah, with the pre-staining, it also helps getting the smaller craters. So here, even craters we see down to almost 0.5 micron in diameter. And this is at optimized laser energies. You can control the crater size with the pulse energy, but here we kind of optimize the energy to still get good signal, and we're still getting easily sub-micron craters. And this works across a variety of different matrices as well. DHE, DMAC, and CHCA kind of holds true. So yeah, I just show this because I think it looks cool. So this is mouse lipid imaging, just it's about a 1 by 1 millimeter area. This is only 2 micron pixel size, just because so we can get a bigger area in a more practical time. So the instrument runs at around 10 hertz. But you can see we're resolving all these individual cells within the tissue. There's a lot of these fine, details, which we're still not really sure what they are, but they're showing up these pink ones with these ceramides. But you can really just see, the level of detail is looking really nice. Again, we can do this for the nucleotides, and I just use this to point out that even with all this pre-staining step, we're still preserving a lot of the localizations, even at the cellular level. So you can see these Purkinje cells here, which are kind of like one of the favored cell types for evaluating spatial resolution in brain. These are really enriched in adenine, for example. So these are all individual cells. These are only about 10 or 20 microns in diameter. There's many, many pixels across them. This is a 1 micron. And then, this other layers in the hippocampus have these different nucleotides. And then, of course, we get all the lipid images as well. So one area we're really interested in exploring this is for doing single cell imaging, but in situ single cell imaging within tissue. So where we know the whole spatial context and the environment around the cells within the tissue. So we've been doing this a lot in the context of MND, motor neuron disease. I just show data here now from just healthy spinal cord tissue, and we wanted to see, can we resolve these individual motor neurons within the whole context of the tissue. So firstly, because we have ablation sizes of around 750 nanometers, now we're doing a 750 nanometer step size. So we're still not quite oversampling. This is the pre-stain image, the optical image. And you can see here's the motor neuron. It's quite chunky. And you can see we get very, very rich data, even at 750 nanometer pixel size. And if we do, this is a PLSA overlay of 3 different PLSA components. And you can see here we can resolve the individual cell in yellow, which has the corresponding profile over here, from the surrounding environment that's made up of different cell and tissue types as well, each having their own distinct lipid profile. So we can push this as well further. So we can go down to about 500 nanometer pixel size. So now this is oversampling, so the signal does drop off a bit. So there isn't as much signal, but now you can still see, we can see a motor neuron, and now we're not even doing a multivariate analysis. These are just individual ion images of 5 different species, where we can also, again, clearly see the individual motor neuron. It has obviously PC34-1 in it. There's these little yellow features as well in the surrounding environment. And then we can obviously pull out the profile of this cell, which is the average now of several hundred pixels, and get the profile of that cell within the tissue. Obviously, we're very keen to see what sort of subcellular features can we resolve with this as well within tissue, which is something we're actively exploring as well. But we are very excited by this to try and even push down to less than 1 micron. So we're doing this a lot for obviously single cells as well. And this just shows the benefit of doing this. So this is kind of 5 microns, the best you could do commercially. And then if you go down to 1 micron, you just start seeing a lot more spatial detail. So even going from 2 to 1. I'll just show a couple of examples. We've done this on a variety of cell types now. So these U2OS cells, we get very rich lipid profiles, especially things like PEs that kind of suggest that we haven't overly fixed the cells, because if we fix too much, we kill all the PE signal. And these are just 3 different overlays of different ion images. This is the optical image. This is fluorescence as well, so we stain with DAPI and MitoTracker. And then this is the overlay of the lipid images with the microscopy. You can see they're really one-to-one. We're not getting any delocalization. We're seeing no lipid signal outside of the cells, and we're even starting to resolve these subcellular features. This is just another example where we can do fluorescence microscopy first. We stain with Nile red to look at different polar, apolar environments within the cell. We do now just the MSI at 1 micron. And you can see we're kind of starting to resolve subcellular features. It's obviously a bit tricky because you've got a 3D cell compressed to a 2-dimensional plane, so you're sampling multiple organelles within a single pixel, but still, at least for a first run, it looks pretty good. And we always like to overlay with the microscopy, so this is the 50-50 overlay in the microscopy of the MSI, and just you can really see that these are really one-to-one, even beyond the whole cell level. That even with all these washing steps, we're still preserving a lot of the localizations, at least on the 1 micron and probably below level. And this is just a zoom of where we see this mitosis event. Just a couple of other examples. These are iPSC-derived neurons. This is a 2 micron experiment. These obviously form these big networks, but I just show this because I like it, because it kind of shows why you want to do single cell. Because there's this one little lonely cell up here that has this lipid at mass 724 that's pretty much completely absent everywhere else. So, I think this is just a nice example of why we want to look at, cell heterogeneity. There's this one separate cell that's obviously very different from the rest. Microglia are a very good cell type. These we see probably the most heterogeneity within microglia. So this is just showing the example. This is 2 micron, but you can see we're getting really fine details within the cells on the MSI level, and we also have the underlying microscopy as well. So finally, just some other work we've been doing. So we're lucky enough now to have this instrument from Bruker, which also allows us to do transmission mode as well, but now combining it with MALDI-2. So this is essentially a timsTOF flex with a different stage and 2 different beam paths, so we can run it as the normal instrument with the reflection mode, or we can open and close certain shutters, and we can bring the laser in through the back and do a transmission mode experiment. So this is the transmission mode optics. We've only had this running in the lab for a few months now, so I'm just going to show one data set which we've just acquired recently. This is a human prostate cancer tissue with MALDI-2 at 2 microns. I just show this just to show that we're really starting to push down to single cell level or beyond, even within whole tissues, and that using post-ionization technologies, be it MALDI-2 or plasma or others, I think is really key here to enhancing the depth of molecular coverage you can get when you're sampling such small volumes of material. So with that, I hope I've gone the right amount of time. I think I've gone over, so I apologize. I just want to thank the group here in Wollongong and some of the funding agencies, and obviously all of our collaborators as well. And with that, just want to thank you again for the opportunity to present, and happy to take some questions if there's still time.
Thank you so much, Shane. I think I'll take over now because this is going to be the last talk. So I'm going to actually also run the question time. And so there are 2 questions already in the chat. One is about after applying standards, can one compare spraying versus sublimating? But I think that you spray your standards, right?
We have to spray the standard. You can't
sublimate them. And then the other one is, this is amazing. What is the imaging speed of this setup? And I think that is related to the transmission mode. So how long it takes, to take the images that you showed us.
It's good. They take a long time. There's no way around that. I mean, on the Pro with the plasma source, because everything's not fully op, it's all kind of bespoke. That runs at about 8 to 10 hertz with the piezo stage. On the new Flex we have, because that's properly integrated, that runs on, say, if you're doing a 1 or 2 micron step, probably about 28 spectra per second. But it still means like a 1 by 1 millimeter area at 1 micron is still 1 million pixels. So it's always, you know, I guess the point is, you know, we choose the resolution we need. That's why sometimes we do 2, because it's still giving us the information we want, and we can just access a much bigger area in a kind of practical time scale.
Yeah, so these were the questions there. I also had a question about the gangliosides that now you managed to visualize so well in the brain. So this has always been something that I think has been missing on our side and our machine. So do you see a lot of spatial patterning when it comes to gangliosides? As I would expect them to be more informative space-wise than many of the other bulk lipids. I mean, is that?
Yeah, I mean, we do see a lot of different distributions for them. They are primarily they're all enriched in the gray matter. But even within the gray matter, like especially even the 36 and 38 one containing species have kind of very distinct distributions, we find as well.
Okay, so thank you so much, Shan, and I take the opportunity now to close this wonderful day together. I mean, I think that it's been great, and I also noticed that you're a Gibson person. This is actually very nice. By the way, so I mean, that said that, you know, I think that we are all happy on how this has gone, and we would like to invite you for the next year to keep tuned. Not only that, but if you want to join the effort, as for instance, Theodore's lab has done last year, if the PhD students that are connected want to participate, this is a purely PhD student run event. We are all here to listen. And so, yeah, everyone is invited to keep this alive and going, and see you in 1 year. Thank you, everyone.
Thank you.
Goodbye.